We are now going live.
Very good morning and a very happy new year to all. And thank you all for being here early in the morning on this cold, very cold day of January. But there is a very warm welcome to all of you for coming and attending to this event today. Just for everyone's information that we will be, this entire program is going live on our YouTube channel. So we need to stick to the timings, and the proceedings of the day would be coordinated by Kathy here in the lab. So, Kathy, can we start?
Good morning, everyone, and a very happy new year. It is the 7th day of 2023, which also happens to be the 7th day of our new director. I would like to welcome Professor Somti with a token on this auspicious occasion. I would like to invite Professor Sapori and Dr. Neeti ma'am to present the token. I'm sorry. I would like to call Professor Somti, new director of ICGEB, for the talk.
Good morning, everybody. On this cold and frigid day in January, I extend my very warm welcome to all of you with the hospitality and the good wishes that all of us have for Professor Sapori. I think we'll have a very warm and very collegiate day. Professor Sapori, it's my pleasure to wish you happy 75th birthday. It's a privilege to have you here with us on this very happy occasion, landmark day. You know, Professor Sapori, I have known since, I can't say I knew him when I was doing my PhD because that was outside India, but ever since I have come back to India in 1993, Professor Sapori has always been there as a guide, mentor, and philosopher at times in terms of the philosophy of science, the way we should do science. He has had a very distinguished career. I think now it is close to 50 years of very distinguished service to science, marked with very insightful papers, first in the area of photobiology, and when he was at JNU, and then subsequently after he moved to ICGEB in the area of stress biology. So he is one of our own, and he has treated me even before I came here as one of his own. So his work has been very insightful papers, always trying to look for something new that is offbeat and not what everybody else is doing. So that's one aspect of his work, the science. He's also been a very able administrator within ICGEB and just across the road to the Jawaharlal Nehru University, which is India's premier university, and he was the he had a very distinguished stint as the Vice Chancellor of Jawaharlal Nehru University. So we have amongst us a person who has accomplished greatly in science and in administration, because not only as Vice Chancellor of JNU, but as the chair of so many different committees in the area of plant sciences and biology, Professor Sudhir Sapori has guided Indian plant science over now, as a scientist for 50 years and as an administrator probably for a major portion of that. Professor Sapori, it's my honor and privilege to wish you happy birthday on my own behalf and on behalf of ICGEB. God bless you, and we look forward to celebrating your 100th birthday here in this hall. Thank you, sir. Have a nice day.
I would now like to invite Dr. Raj Shekhar for his talk.
So first I would like to wish Dr. Sudhir Sapuri many happy returns of this day, and it is a great privilege to be here. And actually 33 years ago, Krishna Tiwari was asking me to come and present in ICGEB, my work which I was doing in his laboratory. But after 33 years I have a great opportunity to come, first time to come and talk here, that too congratulating my supervisor, It is a really great occasion for me. And first, then second is I'm thankful to all of you guys, whoever came so early in the morning to attend this. And later in the day, as we progress, we'll see. But I want to describe two words for Dr. Sapuri. He is the insightful scientist, but a noble man. I'm using this word, is very significant for me in my heart. But I'm going to justify to you guys when I'm going to talk at the time why I use this word. But at the same time, he is extremely superb mentor. One has to have maybe some greatest luck to be working with him for PhD. So that's why I'm going to repeat once again later. If at all I have to do in my future generations, according to Hindu mythology, PhDs, I want Dr. Sapuri is my PhD supervisor. Thanks. And thanks for coming again, and we'll continue this.
It also happens to be the 7th work day of our new director, Professor Somti, and I would like to invite Dr. Neeti and Professor Sapuri to give him a token on this auspicious occasion. I would now like to start with our talks. Before we begin anything, I would like to inform that each talk will include 12 minutes of time, and there will be a timer after 12 minutes which it will start beeping, and for the extension part, the timer will beep after every 1 minute, and then the next speaker will be called. So we begin our talks. The first speaker is Dr. Ananda Mustafiz from South Asian University. He's working on linking phytosulfokine receptor to ABA dependent pathway in plant abiotic stress response.
I would like to add, there are no question sessions. So you just listen, and even in the coffee time you can talk to the speaker directly. Yeah. Thank you.
Good morning, everybody. I'm Ananda Mustafiz from South Asian University. I'm working in the faculties of Life Sciences and Biotechnology as Associate Professor. So I would like to thank Professor Neeti for inviting me and giving me an opportunity to give a talk in this auspicious occasion. So I have joined ICGEB in 2005 as a PhD student in Dr. Professor Sapuri's group, Plant Molecular Biology, and I have done my PhD under supervision of Dr. Snelata Singla Parekh, and it is a privilege and honor that I could work in Professor Sapuri's group and work on glyoxylase pathway, did my PhD, and Professor Sapuri is not only my mentor, he has a huge impact on me as a researcher and as a person, and he will always be a mentor. And the left photograph is on 2015, and this is in 2022 in Kashmir. So happy birthday, sir, and thank you so much for being my mentor for so many years. So I am just briefly, South Asian University is a SARC university which is located currently at Chanakyapuri Akbar Bhavan. It is an international university comprises of all 8 SARC countries, and we are now shifting to new 100-acre land kindly given by Indian government to us in Maidangari. Our campus is made, so we are at transition now. My lab is dismantled and it is being shifted, so we are in the process of moving to the new campus, and we have a PhD and MSc program in economics, legal studies, life sciences, mathematics and social sciences, and I invite you all to the new campus. So my work is on abiotic stress signaling, and abiotic stress, as we all know, impacts all the major crops. About 80% of yield loss in the major crop plants are due to abiotic stress, and abiotic stress is a very difficult trait to work on because it's a polygenic trait, and it is very difficult to understand how the plants are perceiving abiotic stress. So my lab focuses on small peptides and long non-coding RNA currently. So the small peptides, they bind to a specific receptors in the plant, and by signal transducing machinery they will express a lot of stress responsive genes, and eventually the phenotype of stress tolerance we observe. But what are these receptors? What are these small peptides that are still being explored? So we are working on 2 major peptides. One is C-terminal encoded peptide, and phytosulfokine, that is another peptide. I have a small project on both these peptides. So today's talk, I will be talking about phytosulfokine and phytosulfokine receptor kinase. Phytosulfokine is a small peptide of 5 amino acid. It is a pentapeptide, and it is sulfated in 1 and 3 position of tyrosine residues. And the receptor is phytosulfokine receptor kinase. And phytosulfokine receptor kinase has already been shown that it helps in plant. It has role in plant development and reproduction. But its role in plant stress signaling is yet to be explored. So I wrote a small project in BBT and got the project, and where we have proposed to characterize this phytosulfokine receptor kinase. Initially we did a genome-wide analysis, and we have found there are 15 phytosulfokine receptor kinase in rice, and by in silico RNA-seq data analysis and microarray data analysis, and later on by validating by real-time PCR, we have shown that ISPP-SKS 10, 11, 12, 14 and 15, they are highly stress inducible. We all know ABA is a very important stress hormone, and abiotic stress signaling is either through ABA dependent and ABA independent pathway. So when we given exogenous ABA in rice seedling, we have observed that phytosulfokine receptor kinase 13, 14 and 15, they are mostly induced by ABA, and especially ISPP-SKS 15 in different tissues are highly induced by exogenous ABA. So we decided to characterize ISPP-SKS 15 to begin with. We have chosen model system Arabidopsis, and we have created overexpressing transgenic line by floral dip method, confirm this transgenic line. Also we have obtained Arabidopsis tDNA insertion mutants. We have confirmed this mutant by PCR to show them that they are single insertion homozygous mutant lines. When we grown them in the control condition, we have observed that the mutants are compromised in their growth, the cilic length, and the wild type and transgenic plants grown almost similarly, though there is a bit slight advantage in growth and development in the overexpressing transgenic line. Then what we did, we have given exogenous ABA in all these genetic background, like overexpressing lines, two independent overexpressing lines, and the mutant line. What we have observed, these overexpressing lines are highly sensitive to ABA. At 0.75 micromolar ABA and 1 micromolar ABA, they are not able to germinate, this overexpressing line, whereas the mutant lines are able to germinate. So this clearly shows that probably this phytosulfocan receptor kinase, they are working in a ABA dependent pathway. So to show that this Arabidopsis phytosulfocan receptor kinase and rice phytosulfocan receptor kinase, they are able to function similarly, we complemented the mutant line with ISPP-SKS rice phytosulfocan receptor kinase, and we could restore the default wild type phenotype, and that shows that the Arabidopsis and rice phytosulfocan receptor kinase function similarly, especially ISPP-SKS. So in response to other phytohormones like auxin, cytokinin and gibberellin, we didn't find any difference in the growth, seedling growth. So it shows that probably this ABA dependent response is specific to abscisic acid only. So to confirm that, we have shown that cytokinin promotes seed germination possibly by reverting ABA inhibitory role. So we know that cytokinin works antagonistically to ABA. So when we provided 1 micromolar ABA along with 1 micromolar cytokinin, we could restore the phenotype. The rescue of ABA insensitive phenotype of ATP-SKS13 mutant in the complemented line further indicates the involvement of ISPP-SKS15 in development of ABA sensitive response in Arabidopsis. So in response to abiotic stress in the seedling level, we found that these overexpressing lines are highly sensitive compared to the mutant lines. So we have also checked the expression of ABA inducible genes in the mutant and the transgenic background in absence and in response to ABA. We have checked for 5 ABA responsive genes or which are working in the ABA dependent pathway, ABI5, RAB18, RD22 and RD29B, and we have observed that all these genes are highly induced by ABA in the ISPP-SKS overexpressing line and not in the mutant or the wild type background. So that again indicates that ISPP-SKS15 is probably working in the ABA dependent pathway. So we have checked the ABA responsiveness with respect to stomatal closure in the wild type and the transgenic background. We have observed that the stomatal closure is ABA induced. Closure in the overexpressing line were found to be more closed than wild type, while in mutant lines under same condition, which is in response to 50 micromolar ABA. And also OST1 is another transcription factor which is known to regulate the ABA induced stomatal closure. We found that in the overexpressing line, their expression is higher in response to exogenous ABA. Then we have checked the ROS production in this transgenic background in the guard cells, because it is known that the stomatal closure is regulated by ROS production in the guard cell. We have found that in the overexpressing line in response to ABA, the ROS production is much higher in the guard cell and compared to the mutant and the wild type. And there are two enzymes, RBOHD and RBOHF, which are known to be involved in ROS production in the guard cells. And in the transgenic lines, we have found that RBOHD and RBOHF expression is much higher compared to wild type and the transgenic plant in response to ABA. So we have then checked in drought stress how these plants are behaving. So we have given 21 days of drought stress in the overexpressing in all different genetic background. And after 21 days, we have rewatered the plant, and we have observed that these transgenic plants are tolerating drought stress much better than these wild type or the mutant background. So at the seedling level, they were a bit abiotic stress sensitive, but in the when we grow them, germinate them, and then give drought stress, these plants are much more abiotic stress tolerant. So we wanted to check how these ISPP-SKS15 is working in the ABA dependent pathway. So we checked a lot of interacting partners. We have initially screened their ICDNA library, but we didn't get any hit. Probably this ISPP-SKS15 is a membrane protein, and is library screening by we couldn't find. But then individually we picked ABA dependent ABA pathway genes, and we checked for the interacting partners, and we have found that ATPYL9, which is one of the ABA receptor, and its counterpart OSPYL11 interacts with these ISPP-SKS15. We have confirmed this result by BIFC. So in a nutshell, what we have observed is in response to drought, ISPP-SKS15 is upregulated, and also it interacts with ABA receptor. So, and eventually by downstream pathway, it induces many ABA responsive gene, and which will eventually cause ABA dependent pathway trigger, and that's how it is causing abiotic adaptive stress response. Also, we have found that by upregulating the RBOHD and RBOHF, it is increasing the ROS production in the guard cell and causing stomatal closure in response to ABA, and that's how probably this adaptive response is coming. So all these results we have recently published in Physiology of Plantera. So currently what we are working on is a PSKR phosphorylating this PYL9 and 11, because PSKR we know it's a kinase, and what is the phosphorylation site, what is the kinase activity. We have sent this paper to Plant Journal. It has come for a major revision, and these questions are asked by the Plant Journal reviewers. So, and eventually what is the downstream abiotic stress response, that also we are exploring. So I'm thankful to, of course, Professor Sopori and Dr. Snehalata Singh Lapari. They are always guiding me in my research. I'm extremely thankful to Dr. Manoj Prasad, who has kindly allowed me to use their facility and do the BIFC experiment. And I'm extremely thankful to DBT for funding this project, and also South Asian University for additional funding and infrastructure. So this work is done by Dr. Preeti Nagar, who is currently in US doing her postdoc. And bioinformatic work was done by my JRF, Mr. Ashish. And currently my PhD student Nikita and Abhilasha are carrying out this work. Thank you so much.
Thank you so much, sir. Our next speaker is Dr. Rifat John from University of Kashmir, who's working on understanding the response to cold stress in plants from the Kashmir Himalayan region.
Good morning. It's an honor and privilege to be here, and this work, I will not be discussing in detail my work, but it's a kind of a journey I have started and where I am right now. Sir, I owe it to you. Professor Rajshekhar sir said you are a noble man, but to me you are a saint. So whatever I have been doing at Kashmir University, it comes from you and inspiration from you, sir. So when I joined Kashmir University in 2012, since I was working, I had my training in stress biology. I have done my postdoctorate with Professor Sopuri and Professor Ravi Rajan at the South Campus GU. And during my postdoc and PhD also, I was working on heavy metal stress, drought and salt tolerance. And when I joined university, it was cold, and I saw cold tolerance, to understand the cold tolerance in the plants. Right now, for example, we have freezing temperatures. In the March, we'll find everywhere bloom, it's spring there. So why plants are tolerant to the cold? That question came to my mind. Nevertheless, all of us know that cold stress is a kind of a stress which reduces the growth, development, and productivity of the plants. First of all, I was struck by this plant. It is Calendula officinalis. We call it locally. It is called Hamesh Bahar. Means it grows throughout the year. Most of the year it grows, and it is also used for various religious purposes by Kashmiri Pandits, and it's called Bataposh also. So I started with this plant, nothing knowing about it. There was not much literature review. We started with the basic work, like doing the biochemical analysis first. We collected the seeds from the Kashmir University Botanical Garden. These are its seeds, and we tried to grow it in the plant growth chamber, which is not the growth chamber which we have here. It was a customized growth chamber. I had a limited funding, so we put a humidifier, we put the photoperiodicals, timers, and somehow we managed to do this experiment. And we gave different stresses. We grew it at the 25 degrees and then 4 degrees. And every after 24 hours, 48 hours, we took the samples and did this simple basic analysis because I had no lab and I had to start from somewhere. So this work got published wherein we did the physiological analysis. After that, we did the 2D proteomics. I would like to acknowledge here Professor Niranjan Chakravarti. I got INSA visiting fellowship. I worked at ICGEB for 3 months where I learned how to do 2D proteomic analysis. We did 2D proteomics, analyzed different proteins which are getting differentially expressed in the calendula. And we found that most of the proteins which are overexpressed or differentially expressed are related to plant development and signal transduction, photosynthesis, and photorespiration. And but at the same time, I had a lot of issues in characterizing the protein because calendula sequencing is not done. And a lot of questions were asked. Since it was my first work, I was not confident also, I would admit here. So we took another protein from Brassica oleracea. Brassica oleracea is also a cold tolerant plant from the Kashmir region. It is locally called as hawk. It is the only vegetable, above ground vegetable, which is growing right now there. So this protein was isolated from Brassica oleracea and we overexpressed it in tomato. This work started in 2015 and we published in 2021. You can imagine, how many years we had to put in, do simple experiments. Southern plotting, it took us 1 year to do. We finally did it at a hospital, University campus. So later, then there was a maze, there was a congress in Kashmir University, and somebody came up to me and told me, why don't you work on the maize? Now this maize is very unique in the sense that it grows at very high altitudes in the Gurez region. It grows at an elevation of 2,580 meters above sea level. And this maize does not grow beyond that particular patch. It is a landrace which is preserved over the period of time by the local tribals. And the unique part is that normally we see that it is a tropical plant which needs a temperature of 30 to 35. But this maize does not grow beyond 20 degrees. So it is a unique cold tolerant line. The line has an issue that it has a very low yield, but it is very cold resilient. We compared it with Gujarat maize 6. So again, there was not much literature review available about it. No work was done. We started again from the scratch. We assessed it for the various physiological parameters, and then proteomic analysis and infrared gas analyzer we had got at the Department of Botany. So we grew we compared Gurez local with Gujarat maize GM6, and we grew it in the growth chamber at different temperatures. We gave short-term cold stress. Reason being, we did not follow the whole life cycle, because when we got these Gurez local seeds, grew them at the Kashmir University Botanical Garden, they did not give us the seeds. It could not complete its life cycle. It only grew at the Gurez local at high elevation only. But then we gave a short-term cold stress to understand what is happening with the plant. And interestingly, herein we took a different approach. We had got this, I think from UGC, we got this instrument, infrared gas analyzer. So herein we thoroughly tried to understand the photosynthesis of the plant. We took the plant and grew it in the Kashmir University Botanical Garden again, and very interesting study helped us to get this paper published in photosynthesis research. The uniqueness about this paper is the entire paper is revolved around the infrared gas analyzer analysis. We have photosynthetic rate, we have FV by FM ratio, we have electron transport rate, different parameters we studied, and this paper, after many revisions, got published in this journal. And then we started doing the proteomic investigation again. Again, it was very difficult to work with the protein of maize. It's very difficult. We had to run many programs. This is only a brief overview how we analyzed, and then we did this MS analysis. This is just one of the screenshots. And herein we could find many interesting results in this maize. We found that there is nodular-like protein. Usually there is no nodule formation in maize, but we found this this protein is getting overexpressed. And similarly, there are these threonine dehydratase biosynthetic chloroplasts. They have not been reported so far, palmicylated protein. So, but now we are going to characterize them. And we started doing the transformation in the maize. And believe me, it is very difficult. For past 1 year, we are trying to do the transformation of maize, and we are reaching nowhere. And now we are thinking to do it in the Arabidopsis. So these are the differentially expressed proteins from the maize-6. Again, these are the proteins. In red color, these are the proteins which are not reported in the literature yet, and now we are going to characterize them. Now, another thing is that cold stress any other stress does not occur in isolation. The stresses, there are multiple stresses in the plant. Now, this when there is overwintering during December, January, and Feb, this cold stress or exposure to the cold is very important for the flowering in the month of March and April. Now, there is not change in the temperature, there is shift in the photoperiod also, because we see from December end towards the March, there is shift from short day towards the long day periods. So that is why the flower development or the plant development is not related to the change in the temperature, but also in the photoperiod also. So what we did is we took the brassica rapa seeds, we grew them in the month of October, then in outside, and then we took the 3 phases. This is after the germination, this is vegetative, this is pre-flowering, and this is the post-flowering. And we compared it, we did the microtome sectioning of, and then there are many interesting results which have come up. This is still going on. At the same time, we did the transcriptomic analysis of these 4 stages to understand how vernalization affects the brassica rapa flower development. And apart from the regular genes which are involved in flower development, we have found a couple of heat shock proteins which are related to the flower development in the brassica rapa. Another project which I got is how plants are adapted to different altitudes, because as there are some plants which grow along a different altitudes, so they face that altitudinal stress. Now, herein we took the approach of microbiome, because earth to a plant is like a mother. Plant has a very intimate relationship with the soil, and it's very important to understand that microbiome has definitely a role in this stress tolerance. So the idea of this work was that both this corn, maize, which I said Gurace local, as well as trifolium, that is a leguminous plant, this grows at various altitudes. We went to the different altitudes. I went with my students and did the collection. This is KUBG's Kashmir University Botanical Garden, then there is Convar, then is Tangmal, Gulmarg. In fact, this maize grows above also, up to the Gurace. But we could find that this trifolium grows only up to the Gulmarg, 2,800 meters above sea level. And we did proteomic analysis, and we found that there are 108 proteins till now. They increase significantly in abundance in cold acclimated red clover populations, and they were assigned to response to stress somewhere related to carbohydrate and energy metabolism, amino acid metabolism, signal transduction, molecular chaperones and protein folding, transcription and translation, and metabolite transport. Many groups of proteins showed largest increase in abundance in response to the cold, where proteins related to stress response category, vegetative storage proteins, KS dehydrins, and cold attenuation specific proteins. And we also saw heat shock proteins, also molecular chaperones in red clover or dry folding. Downregulated proteins belong to the category carbohydrate and energy metabolism, signal transduction, amino acid metabolism, stress response, and translation. And then we try to understand, this project is still going on, it is in the third year, this project, this is core grant by SIR. And here we try to understand this endophytic and soil rhizosphere. We saw that there is a steep decrease in frequency, density, and abundance from Tangberg, and then again there is increase. So this is not all the data. This is a small one of the pictures from this data. This work is still going on, and this is my research group. She is Nabiha, Wasim, Tahira, Wasim Yusuf, Reis, and my lab has been generously funded by SIR INSA because I was working as INSA young scientist at Professor Niranjan Chakraborty's lab. That is why I'm acknowledging it here. And Department of Biotechnology, they funded my first project, and it was when I first time got the distillation unit and CSI. Thank you so much for your patient listening, and comments, suggestions, criticism is most welcome. Thank you.
Thank you so much, Rifat. We don't have a question hour or a question answer session after each talk. We'll have enough opportunity for to interact with the speakers during tea and lunch. We've kept longish time durations for interactions there. And can I now request Professor Sapori to please felicitate the speaker, and also Ananda, we're sorry, I think the cold has slowed down our pace, so please felicitate.
I would like to add a small incident. When I joined Kashmir University, sir may not be knowing, I called him up and told him, sir, I got appointed at Kashmir University. He was happy, and then he told me one thing, and that has become mantra of my life. He told me, since you have joined university, do one thing. Don't leave your department before time. Even if you don't have work, stay there. Because it will become habit really early. So since that day, I stayed, during my prescribed office time, I won't leave. He told me, even if you don't have any work, read paper, do something, but don't leave department because it will become a habit. So thank you so much, sir. You have not taught me the science, you have taught me the art of living words. Thank you.
So we all know Richa, she's coming in as the next speaker, and Richa, why don't you just speak one line?
It's indeed a very warm morning on a very cold day, contrasting words. To be felicitated by Sir on his birthday, I think that's ironical. What we want to say here is, maybe have a similar occasion as Dr. Sonti was saying on your 100th birthday as well. I would be talking about my journey. As Rifat said, we are talking more about our journey than about science. With perspective from allelic variation in plant responses to abiotic stresses. I have done my master's at JNU. Sapuri sir was my advisor. I submitted my thesis on P and calcium in the year 1999. And 2000, I joined the Millennium batch of PhD students at ICGEB. I worked on pearl millet. This is not the main content of the thesis, but while we were working, one day sir was asking, "Na, ऐसा क्या किसी और में भी होता है?" I said, "Why do you keep talking about अब pearl millet में कर लिया तो अब P में करेंगे?" This conversation happened, and he said, "I think like a botanist, no, and you people are only bothered about one molecule, one pathway, one target." And in that background, we isolated isoforms of vacuolar ATPase-C, and there was a real-time PCR machine which was assigned to Dr. Suresh Nair and me too, and that machine we took to understand how these three isoforms, which are different alleles of the same gene, behave. And interestingly, we found a novel isoform which was isoform 2, which was only induced under stress in sodium chloride and calcium chloride treatment. It is not inducible in shoots at all. It's root specific, but under stress condition, it's expressed in the shoots. So with that naive question and even very simple experiments, I did a brief stint with Dr. R.P. Sharma. He was, that time he had just got a tilling population in tomato. So the first introduction of what the allelic variation might look in a population was given to me, PhD being in molecular biology, and with that background in 2005 I moved to Cornell, this is 2006, I moved to Cornell University, and this happened to be Department of Plant Breeding and Genetics, not biotechnology, not molecular biology. And one of the first assignments that was given to us was a bigger question. There was this person who was working, evaluating the grain arsenic content. The student was a joint student of Cornell and Bangladesh Rice Research Institute, and they wanted to know whether the lines released by BIRI were sufficient to combat the arsenic contamination problem, which is a predominant problem in Bangladesh. And upon evaluation of 38 varieties, this was our component. We realized there is no significant genetic variation, and novel genetic variation will have to be introduced in the breeding program if they were looking for grain arsenic tolerant genotypes. This subsequently was followed by the continued learning as to what does rice look like. Does rice have a population structure? Does that population structure come from inherent allelic variation? Does it have anything to do with domestication? So the entire postdoc journey was not on molecular biology per se, but trying to understand population structure in a crop, rice. We landed up designing the first SNP panel in rice for which was evaluated on around 400 rhiza sativa lines, and this led the foundation for the later chips that came for the genotype-phenotype platform association in rice, where the rhiza sativas and the rufibogons were brought together with a set of 44,000 SNPs. There's 1536 SNP. And so what does this tell us? Classically, rice has been evaluated with use of RFLP markers, AFLP marker, SSR markers. So the SNP data did not give us something new, but what it just proved was there was a way back paper using different markers, chloroplast and microvoltio, that there is a distinct population structure in rice, and the SNP markers proved that. Our rufipogon complex analysis also, this was done by genotype by sequencing, also proved that there is a subpopulation structure that exists in the samples that were evaluated from multiple countries, wherein you could have an indica type subgroup, you could have an aus type subgroup, and you had a admixture of rufipogons coming in. With that background in mind, in 2009, I came back and joined Central Agriculture University at Meghalaya, which is one of the constituent, and this was College of PG Studies. The The idea was that the soil is acidic. 74 million hectares of soil in India is acidic in nature. In African region, it's around 35%. When the soil is acidic, not only it leads to all kinds of mineral deficiencies and toxicities. You have iron toxicity, you have aluminium toxicity, and you have phosphorus deficiency. These are typical symptoms of how toxicities with respect to rice look in the field. Again, the same question was asked, can we take what we know from Arabidopsis and impose it on rice and use that information to have P deficiency tolerance mechanism understood in rice? Now, with a slightly better understanding of what the allelic variation might be, if you can just have a look, the genome that is sequenced is a temperate japonica, and most of the tolerant genotypes come from the house clade, whether a classical example is Kasla. So rice genotypes are diverse and distinct, and there is a genetic structure that exists. With that background and the Arabidopsis story and the rice story which was emerging, which was PUP1 PS-stol, we asked the questions, is root structure dependent on genetic background? If yes, then alleles of key genes be associated with genetic background. If no, then alleles if found should be functionally similar. There were many unasked questions in the PS-stol story as well. Parallelly, we were looking for genotypes which were novel. That means they did not have the reported loci. We screened the germplasm which was available with us, and focusing on the northeastern lines, we found rice donors which could be used in the breeding program, both which had PS-stol1, which did not have PS-stol1, and which had a novel haplotype for already reported genes. This was followed by a transcriptome analysis. We focused on roots only to see what happens when two contrasting indica rice genotypes, one Sabhagidan which has PS-stol1 and one Chakha which does not have PS-stol1, how do they behave? And we could find mechanistically different pathways wherein the Chakha, which lacks PS-stol1, uses its internal phosphate to remobilize energy and respond to P deficiency tolerance mechanism. This was followed by targeting genes which were unique to either of them in terms of expression for marker design, so that these markers could be used in breeding programs as well. Parallelly, we were part of the national program where the rice germplasm from India was genotyped so that we could have a mini core. So 6,912 rice genotypes were genotyped, and including the germplasm from northeastern India, as a result of which you have a rice mini core available of 98 genotypes which can be used to ask n number of questions. We were still asking the same question, if P deficiency tolerance is a quantitative trait, what should be the approach? Right now we think population improvement could be one of the best ways to addressing that. The other thing we were also asking is, iron toxicity QTLs are reported but underlying genes are not known. P deficiency mechanism is very well reported. Can we do something about it? Simultaneously, because aluminium toxicity is also a problem, we had identified genotypes based on upland phenotype, and they were characterized as well. So throughout this, the genotypes which are adapted to acidic soils of northeastern hill region showed a mechanistically different mechanism as compared to rice genotypes which were sequenced or available in the databases. We landed up identifying gene members which were playing a role in P deficiency tolerance. The low temperature story, to bring back what Rifat was talking about, was not that interesting in terms that when we did allele mining for DREB1 and DREB2 in diverse genotypes, we found that it's a highly conserved pathway for low temperature. We could not find allelic variation which could be targeted and brought into the breeding program. However, the low temperature germination QTL there's a 71 base parendel reported, was found in genotypes which had never experienced cold at all. So parallelly, the campus was developing. We were seeing Meghalaya, that's the pitcher plant on the left, and having colleagues and students and enjoying the local hospitality as well. We landed up narrowing down a novel region of rice of 1.84 MB region. We know there are allelic variations there. These would be part of our next phase of functional validation by using gene editing approach. Recently we have found that one of the iron deficiency tolerant gene regulator, negative regulator, IRO3, has indel in the 3' UTR, which correlates with higher yield under iron toxic conditions in the acidic soils of Meghalaya, and it works across 43 diverse genotypes as well. So this brings us back to the question that all this journey has led to pose. We have been focusing on a molecular toolbox, putting our signal transaction information into there. We've identified multiple candidates, but many of them are either not validated in the field, the breeding cycle is too long, the yields are stagnant, and diseases are emerging. Where do we go from here? Haplotype, that means combination of desired alleles, offer multiple targets, knowing the breeding value, and pangenomics would be the way. Rice serves as a very good example where superior haplotypes for many of the polygenic traits are available and can be incorporated into breeding program. So the next phase of the story would be genome editing, wherein can we understand the trait, take the allelic variation, and do a functional validation. One example being, this is a color pathway which works across cereals. This has been validated by us at the transgenic level in 2017 with editing by different groups. But the same pathway branches out to lignin pathway, which has a role in stress biology, especially biotic stress tolerance, is also emerging. So you're talking of a similar pathway. The question remains the same. There are common molecular players. Can we do a trait discovery wherein we can have alleles which can be used for editing? In cases of, now I'm talking about wherein this work would fit in with the equisat work. In case of chickpea, sorghum, pigeon pea, pangenomes are available. What is needed is to identify a functional allele which would be fitted into the breeding program. I would like to summarize that this journey is still ongoing. We are still learning many components of the traits, trying to decipher what the alleles are doing for that trait. And this would have not been completed without all the mentors who are there in the room right now. I can just name a few. Sir has been a major influence in terms of how do you cut the noise. This is an amazing quality of Sir. So many things may be happening, but when he is focused on a problem, everything else goes into the background. I hope I learned that, and along the way a bit of patience, which he has immense store of. As a mentor, I think one thing the students really want from you is that unflinching support and the patience to listen to absolutely strange and bizarre questions. He has never even once said, oh, this is a crazy or a stupid question, why did you ask that? So I think that is what I have learned in so many years. Thank you all for very patient listening. Thank you so much.
Thank you, ma'am. I would like to call Professor Sapori to give the token. Our next speaker is Dr. Shigaki from University of Tokyo, Japan, who is working on on-site detection of New Panama disease of banana using the LAMP-based kit.
Thank you very much for a kind introduction, and I feel very honored to be here to celebrate Dr. Sopori's 75th birthday. Very happy birthday to you, Dr. Sopori. Thank you. And okay, and before I go on to my talk, you see, I need to explain why I'm associated with Dr. Sopori. It dates back to 2007, and okay, my wife is from Gujarat, And but I was in the U.S. and so I needed to bring my wife to the U.S. But so there's a visa problem for Indian citizens, and I could not bring her. So instead, I decided to come to India to spend 1 year, and I needed something to do as a scientist. So I asked my friend, Dr. Rajaseka, and then Dr. Sopori helped me to find workplace. That was the Indian Institute of Advanced Research, IIR, in Gandhinagar, Gujarat. So that happens to be my wife's hometown, Ahmedabad. So it was very convenient. So I would like to thank Dr. Sopori for this, giving me this opportunity. And as a result, I was able to spend 1 year with my wife. Anyway, so this is IIR. This was a premier institute, and it was very well equipped. And what I did in this institute was DNA barcoding. That was back then, a little bit new. So I decided to take up this technology, and I chose acacia. That was an important tree for India. So we collected many samples of acacia, all kinds of different acacia, mainly in the state of Gujarat, and analyzed DNA region. And so we were able to publish one paper while I was there. Okay, so, and today's topic is not this DNA barcoding of acacia, but about bananas. So, and it's India is a banana country, I know that, and I enjoy eating bananas. And but then, okay, do you know this banana variety called Gros Michel? And probably very few people have eaten this, and it's extremely tasty. It's creamy and sweety and soft. So, but this went extinct, actually. So extinct means extinct commercially, not biologically. So it's we can still find some Gros Michel banana plants somewhere. Yeah. So I happened to be there. Actually, it was in Papua New Guinea. So, and there was a banana gene bank. So one of the trees was Gros Michel. So I was able to eat whenever we were able to harvest this, and I enjoyed it. Of course, it's much better than the Cavendish bananas that you can buy from stores. And okay, so let's go over the history. In 1950s, so this Gros Michel, very tasty banana, was the dominant banana varieties back then, but it was, it had a fungal problem, infected with Fusarium oxysporum, forma specialis cubensi, FOC. And this, as I told you, it led to commercial extinction, because they were able to harvest so little, and it didn't make any sense to sell it commercially. And then Cavendish banana that you can find in stores nowadays was identified as FOC resistant, and became the dominant variety up until now. And but recently, a new strain of FOC called Tropical Race 4 appeared. We call it TR4 for short, and that could destroy this Cavendish bananas totally. And okay, the symptom is just like that. It's yellowing of leaves from outer appearance. And the history of TR4 appearance is that it was first found in Taiwan in 2006 and spread to Indonesia, China, Malaysia, Australia, and eventually Philippines. Philippines is the largest exporter of bananas. So now you see they have this company called Dole, having large plantations there, and they have a serious problem. they cannot supply the market, banana market, with enough bananas. They are losing money. So it's a very serious, real problem. And in 2013, this TR4 strain spread to Jordan, Vietnam, Laos, Pakistan, and Lebanon. And in 2015, TR4 reached Africa, Mozambique, and eventually in 2018, it was reported in India. So I don't know much about this Indian occurrence, but I heard it's from Bihar and Uttar Pradesh, and also in Gujarat. So at this time, I don't think it's big concern, but it can become a big thing in the near future. And then it went to South America in 2019. It went to Colombia, and so this Indian case is particularly of concern because India, as you know, the largest banana producing country. Philippines is the largest exporter, but bananas are domestically consumed in this country. So it probably it's a more serious problem because it's food for people, everybody's. Okay, and the symptoms are similar to other diseases like a phytoplasma. You can see yellowing leaves, and if you cut the pseudostem, you can see the brown ring. So yeah, that's a typical symptom. But it's, because symptom can be from anything, accurate identification is required. It has to be, if it's TR4, it has to be fusarium, and some accurate detection tool is required. And at the same time, this early detection is a key because there's no known good resistance against TR4 at this time. So only thing we can do is to stop the spread by quarantine methods. And for that, we need some good detection method. Okay, so then we are introducing this lamp. Maybe you have heard of this during the COVID time. The PCR is most popular way to detect COVID virus. But lamp is also used, and it's sort of like better, more accurate method. But it's not as widespread as PCR at this time. LAMP stands for loop-mediated isothermal amplification. So the key word is isothermal, so same temperature. And you see, unlike PCR, which requires 3 different temperatures and cycling of temperature, this isothermal method, like LAMP. There are many isothermal methods of DNA amplification, but LAMP is probably the most popular. And because of that, we don't need a thermal cycler. We can perform it in a single temperature, like 65 degrees. And okay, I will not go into this biochemistry of how the DNA is amplified, but it's a totally different biochemistry than PCR. But result is the same. You can have this amplified DNA so that you can visualize, but you don't need to run a gel, and you can visualize it using this color. You see, and okay, sensitivity can be 1,000 times better than PCR. And okay, we have this dry kit. The advantage of this lyophilized kit, the reagents are all mixed so that you can reconstitute with a buffer, and you can use it in the field itself. You don't need a laboratory to perform this. And you can take it, and you don't need to store it in the cold. You can carry it in your pocket, probably not in the pocket, but maybe the bag, and take it to the banana field where this TR4 happens. And okay, so this is not sorry, but this is not the banana testing. This is the coconut testing, but based on the same ideas. The main thing is that we can do it in the field, so that's a great advantage. So these pictures show this coconut phytoplasma detection actually, but we want to make a kit for this banana disease TR4, New Panama disease is called. Okay, so and this you see this lamp kit was tested first time. Lyophilized lamp kit was tested for the first time in South Pacific country called Solomon Islands, and somebody saw this dwarfing leaves of sweet potato, and it looked like phytoplasma disease. And we had the phytoplasma detection kit, lamp kit, and so we decided to test it. And then out of 6 samples, 4 turned positive. The left 6 tube samples, and you see this green color means positive. So it's very clear, and you can do it in the field itself. I think it's an advantage. Okay, so we found this paper published from Wageningen University in the Netherlands, and they published primers for this TR4 strain, and we decided to use these primer information to develop this the lamp kit for the banana disease. And okay, and it's ready now. It's ready now. Prototype is ready. So, and it was made in our university and a company called Nipponjin, so in Japan. So we brought it here, and okay. So the lamp kit was only tested in the laboratory. So maybe it's better to test it in the field if possible. And we don't grow bananas in Japan, so we have to go somewhere in a warm place, like India. And okay, kits for other plant diseases can be also developed, not just bananas. So this method is a very efficient way, an easy way, a quick way, more sensitive way to detect any plant pathogen. So yeah, so this maybe a TR4 is successful, we can develop more kits for other diseases as well. And as I mentioned, it has many advantages over PCR. Of course, PCR can do other things, but as far as pathogen detection is concerned, maybe a lamp is a better and efficient way to do the job. Okay, and I would like to thank, first of all, Dr. Sopori for giving me this opportunity to speak in this premier institute, and also Nipponjin company staff and University of Tokyo staff, Dr. Namba, Majima, and Dr. Miyazaki, and also the people at Solomon Islands Ministry of Agriculture and Livestock. Yeah, thank you very much.
Thank you, sir. I would like to invite Dr. Sapori to give the token. Our next speaker is Professor Pradeep Agrawal, CSIR Central Salt and Marine Chemicals Research Institute, Gujarat, who is working on halophytes, unique source of salt-tolerant genes and transcription factors for developing future climate-resilient crops.
Good morning. It's my great pleasure and honor to be here on the occasion of one of my guru's 75th birthday. It's rare opportunities for the youngsters and the workers to present in such a situation and speak also. So sometimes you are afraid also speaking in such a in front of the people who taught you and who is one of your guru. And just for a small, like when I came back from US after spending several years in Holland, I met Sir, went to chamber. So now what to do? Sir said, okay, come, don't worry, we will do something. You have done good, you have a good experience. And then he took me to Dr. M.K. Reddy's room. And both of, like the gentleness of both of the people made my life easier and working further in my life on the plants. So here, like I started my work in ICGEB on the sanitary toilets. And then I moved to Gujarat. Gujarat is a nice place. It's quite pleasant right now, but Niti called me in this chilling winter in Delhi. So I'm working there in the Central Salt and Marine Chemicals Research Institute. It's a CSR institute. And the name is coined by Central Salt. Coastal area has a lot of salt pans and a lot of salt is produced there. And you eat, I think, a lot of salt from that place. And there is a coastal area where lovely plants grows, and very few people knows about those plants. So these are the plants which are unique among the different plants, and they consider around 1% of the total plant kingdom. It's a large number, but they have a unique property to tolerate. So some of them basically accumulate salt, and some of them degrade or secrete salt. And this is a very unique property in these plants. Like many plants have trichomes and these glycophytes has trichomes, but they cannot secrete salt from their trichomes. So these halophytes basically, they have a salt bladders or epidermal salt bladders which are modified trichomes, and they release salt from this one. So generally their property or genetic unique physiological mechanisms that they do not store more sodium chloride in their cytoplasm, so they excrete it out. So plants which are like glycophytes, their osmoticum is different. They produce the osmolytes, but these plants take the advantage of sodium and chloride to make osmolyte for them. So in one word, they have less energy consumption, like less ATP consumption compared to the glycophyte plants. So because of this reason, I attracted with these plants and working on this one. So Gujarat has approximately 18 different halophyte plants. I collected these plants from different area of Gujarat, and I tried to see that which one is salt accumulating and which one is salt secreting, because both the properties very, very different physiologically and how they secrete out. So then I developed this halophyte park, and then Dr. Harshvardhan was, Honorable Harshvardhan was our science minister. He also visited one time to Serai, and he visited halophyte park and to see those uniqueness of the plant. So he took a lot of interest in that, and further, this is, I'm just giving you one or two slides for about the general idea. Then we are also involved in the Salicornia cultivation. Salicornia is a plant which accumulates salt in its pith region, and approximately 20 to 30% sodium chloride is there in the Salicornia plants. So our institute has produced the salt from this plant. It is known as herbal salt, as it has a low sodium chloride, means 10% potassium chloride in this plant itself. But we are also modifying the salt by putting the potassium chloride from the algae Kappa ficus, which has high potassium. So this plant have the very unique life cycle. When they basically go to the older age or after completing 7 or 8 months in the field, they turn radish and mature their life. So this plant is basically cultivated right now by my group in the Gujarat coast to help the local farmers, because local farmers does not have any kind of the monetary help because they cannot grow the crops. So they are doing basically cultivating this crop for the salt industry. So now coming to the molecular and biotechnology work of the Salicornia and Aeroropus, which is my original field. So we have started this work earlier, long back, but recently with the Professor Sonti's former group of CCMB, we did this genome sequencing and genome analysis of this plant. So we find out that the genome size of the salicornia is approximately 100 MB more than rice and other glycophytes. So this is one of the important property of the salicornia. So then we are right now doing the whole genome sequencing as well as transcriptomic profiling of the salicornia to see why it accumulates salt in the pith and why other plants basically secrete the salt. So in our journey, this work I started long before with collaboration of ICGEB. We did cDNA subtractive hybridization and isolated large number of genes from that, and we characterized the genes like antiporter genes or transcription factor. So to understand and functionally validate these genes, like how they are basically different from the normal glycophyte plants and how they are inferring the salt tolerance in these plants. So several genes are validated during the course. And then this is one of the very interesting plant where you can see Aleuropus lagopyde. It's a monocot plant. So Aleuropus lagopyde excretes salt from the crystal from the leaf surface by the help of the salt bladders. This picture in the, like you can see in the middle, like white crystals, and Gujarati call it, recently my student call it diamond plants. So there's it's like releasing diamonds. You see in the microscope, you feel like crystals. Then we find out that what these crystals are about. Then we did SAM EDX and find out that major portion of this SAM EDX property is sodium. So but the uniqueness in this plant is that the number of the crystals get increased when you increase the concentration of the sodium chloride. So Dr. Sonti always asks me that why they are basically excreting salt. So is there any property in these plants they are excreting salt? Till now, in Arabidopsis, 40 genes are isolated which are basically involved in salt secretion and developing the modified EBC, epidermal bladder cells. But in Hyrophytes, no genes is still reported that which genes are basically responsible for forming the EBC or putting the salt in the EBCs and then releasing further. So we are doing right now the whole genome sequencing as well as transcriptomic data, detailed transcript data to find out those genes and find that how these genes are basically involved in doing the salt secretion. Only on that time we can basically transfer those genes and making the trichomes of the glycophyte plants towards the EBC, because EBC is basically enlarged 10 times enlarged epidermal cell, 10 times enlarged, and it can accumulate 1000 times more sodium chloride than compared to the glycophyte epidermal cell. So then we have analyzed some genes like RAP7, which is endocytosis, exocytosis involved, and several transcription factor from this plant to understand the functional validation. So this genome size is almost same of the Aleuropus, and then we have analyzed the transcriptome data that how many genes are upregulating and downregulating in these plants, and then further this is the shoot and root, because both shoot and roots are quite unique in Hyrophyte plants. Some plants are basically prevent the salt entry at the root level, and some plants are basically which have like salt is entered in the leaf part, so how they are managing from the cytoplasm, that is also helping. So then we are characterizing or basically classifying large number of genes in different classifications and looking after some genes, and luckily we got around 17 genes which are involved in the EBC conversion into the epidermal bladder cells formation, trichome converting into the EBCs. So now we are characterizing those genes, how they are helping. So further we are transferring 1 or 2 genes into the CSEM. CSEM is one of the important crop, and DBT rejected my project because the regeneration protocol is not available. Those who work on the system, the regeneration is very, very difficult, and Dr. Grover is also nodding me, yes, you are right, Pradeep. So we developed the regeneration system in the SESEM, and we published this paper, and this is the SESEM property, why SESEM is important. India is largest hectare area in cultivation, but the lowest in the production because of many problems in SESEM. SESEM has large problem of the pesticides, infestation as well as salinity problem. So we developed 2 type of protocol. One is in planta transformation, and here we tried different stage of the embryos or different stage of seeds for transformation, and then we got this much of result. and all my SESAM friends in India, they surprised that we got at least this much of result. We have right now 17 plants out of 1800. You see the number of plants, 1800, 1200 or 2600. So we tried approximately 15,000 explants till now, and right now only have 6 plants in the greenhouse after transformation. So thank you. This is the entire journey of teaching of SAR, and to help me standing here and speak in front of you. So thank you so much.
Thank you, sir. I would like to invite Professor Sapori to give the token. Our next speaker is Professor Syed Hasnain from IIT Delhi, and he's going to present on my academic journey from green algae to plant storage protein to baculovirus to mycobacterium.
Very good morning in this very chilly morning. It was difficult for many of us, I'm sure, to come here. First of all, a very happy 75th birthday, Professor Sapori. Professor Sapori and I joined JNU. Of course, I joined as a master's student, and he was an assistant professor back in '75. A year ago, I think you joined JNU. But we were more like friends. Those days, JNU was very different, and we have cultivated that relationship for long. I initially worked on, he was my mentor, though not directly PhD with him, but with Professor Kailash Upadhyaya on Chlamydomonas. So when I was asked by my good friend Rajshekhar that I should be speaking, I said, this is a plant meeting, what will I be doing? So I thought I'll give you a flavor of what we did earlier in plants. In fact, my last work in plant was in a PNS paper that has been cited 350 times. This is a rice basmati DNA typing, and that was done when I was working at the CDFD, Center for DNA Fingerprinting. And so I still have some connectivity to plants, and listening to you today, I realized what kind of a broad canvas under which Professor Sapori used to supervise his people. It was great. I've known him, we have been together in a few committees, but I thought I must be here today a whole day to be with you, sir, to wish you and also to get a flavor of the kind of influence you have been able to make in the generations of ours and younger. Thank you so much, Raj, for inviting me to be here. If I can move slides from here. Yeah. Change is the only constant in life. I firmly believe, and this is very true. I made this statement some time ago, and somebody quoted me and said, this is a wonderful statement. I said, yes, it is indeed true. Starting from life as a Chlamydomonas in Hardy, we published a lot of papers. That was when I was working at the Jawaharlal Nehru University, School of Life Sciences. Of course, some of the work continued beyond my stay at JNU. Then I moved to herpes simplex virus as a postdoc, where we published, and also Aclea ambisexualis, and then to sorghum storage protein gene with Tim Hall at the University of Texas, where we did molecular biology of the storage protein gene caffeine. I stayed there only for about a year, and not more till because I got a job at NII, National Institute of Immunology. And after I moved to the Institute of Immunology in New Delhi, I thought I should not be continuing my work, what I was doing earlier, but let me do something brand new. And that's where I started working on the baculovirus expression vector system. And we developed this baculovirus expression vector system for the first time in India. We cloned and expressed maybe about 50-60 genes of biomedical importance, including vaccines, including not only in the cell lines, but also in the insect larvae. And then subsequently I moved to Mycobacterium tuberculosis, and there was reason for moving. My lab has been growing very steadily, and the last time when I was in the baculo expression group, we have about 30-40 people in the lab. Impossible to have so many people in a small lab, so I got a deal from my then director, Dr. Sandeep Basu. I'll give you a brand new lab because ICGEB is moving away from that campus to this new campus. So that lab is being vacated. So I'll give you, I said, that's wonderful, but you have to move to tuberculosis because TB is a big grant we have received. I don't want the grant to go back to DBT because the PI has moved to industry, and I don't want the grant to move to industry. So would you be willing to work on TB? I said, I don't work on TB. He says, no, but you have been changing your area, so move to TB. TB is something where you can benefit people. I think that was a very good suggestion, and I really would like to thank and place on record the suggestion made by Dr. Sandeep Basu. He asked me to switch to TB, and TB has been really very productive. We have some important decisions, important things that I'll give you a small flavor, not much though. A baculovirus, as some of us may be knowing, it's an insect virus. It's known as nuclear polyhedrosis virus. It has, is it a pointer there? Yes, this is a pointer, no? So it has this is a caterpillar that actually you can grow it in the lab, you can like greenhouse. And this is in the hindgut, actually in the midgut, it produces the virus which you see here. It's occluded as a occlusion body, and the occlusion body is made because of a polyhedrin envelope, and polyhedrin is the largest expressed. More than 50% of the protein at that point in time in the cell is polyhedrin, and it is one of the strongest promoter. I don't know which is the strongest promoter today, but that time it was considered to be the strongest promoter known in biology. How this virus makes this protein in such large amount. So we not only worked on trying to express proteins both in the caterpillar as well as in the cell line, but also asked some very fundamental questions about how the polyhedrin transcription goes on. And this was a very primitive model when we started working. We knew that there are some upstream activators, there are some promoter binding proteins, there is an RNA polymerase. Is it a host specific or is it a viral RNA polymerase? And there are also viral proteins. The sequence, of course, was known completely. It's a small virus, 138 kV virus. So it was known, the polyhedrin gene promoter was known. So we started working on it, and I think in the next 4 or 5 years, or maybe more, we could completely show how this transcription was progressing, and we published a lot of papers, including in JBCs and many other good top journals. And we showed that there are late expressing factors, the left proteins, there is viral RNA polymerase, and most importantly, there is a nucleating protein which we called it as polyhedrin promoter binding protein. And this is a host protein. This protein actually is recruited by the virus to the promoter, and then it in turn recruits other proteins which are some viral factors, some other host factors, and we also identified an upstream enhancer. We call it HR1 enhancer sequence, homologous region sequence, which is present quite upstream. And this HR1 we found we have already patents and it's been licensed. This sequence is known to be enhancing expression. So in a short time we could in fact we sent this paper to Cell, and Cell said that it took a long time. They finally sent it back asking us to do a lot of experiments, because in this paper we also showed that there is a linking of transcription and translation in the insect cell system. And we realized that maybe we may be scooped, so we decided not to revise the manuscript. We split this into two. One paper went to PNAS, other went to another journal, and we managed to publish, and that was a good thing because at least we now have, some people do know how this promoter is done. We also worked on a protein known as the P35 protein. It's an anti-apoptotic protein, and this is a work that we published, and we found that this protein is an anti-apoptotic protein. It acts as an initiator phase, effector phase, and an execution phase. It's a little complex cycle, but then we could understand and show how this virus, and this was my first exposure to host pathogen interaction, which means if you have a pathogen such as baculovirus in an insect cell host, how does the host interact with it to ensure that the pathogen survives? And this was eventually the host has to be killed. And this protein has a very important role to play in host pathogen interaction. It's an anti-apoptotic protein. It favors the host, favors the pathogen, the virus, because it's anti-apoptotic. It doesn't let the cells die of apoptosis, because then the virus will also be cleared. So that's one of the things that we did. Having done this, we got very well known that we have a wonderful system to express proteins, and we got a request from a group in CDC Atlanta. And they said, can we make a malaria vaccine in the baculovirus system? And we did not know much about that at that time, about malaria vaccine, because it was something brand new. But what we did was, there were a lot of things known about what are the major epitopes when malaria parasite infects, mosquito infects us and we get malaria, there are some dominant epitopes. So we thought, why don't we stitch these dominant epitopes, do a reverse translation, stitch these epitopes in the form of DNA and put this into the DNA for a string of beads structured in the baculovirus system, and express this in the baculovirus system. We did not know at that time the rules of protein folding, and I think not knowing the rules really helped us, because it worked. The protein was expressed, it gave huge amounts of protein, and actually it was a wonderful thing that I realized. According to accepted principles of aerodynamics, the bumblebee cannot fly at all. Aerodynamically it's not possible, but the bumblebee does not know aerodynamics, and it flies anyway. So we thought, if we don't know the rules, let's make use of that and try doing it, and we were successful. Successful in the sense of making this protein. This paper was published in PNAS. It was very well received. This was subsequently tried. I do not know what's the status right now, it's not my area. But this was in falciparum malaria. We have vivax malaria, which is more common, and I think more people now today are working on the vivax malaria. But this paper was covered in Star, in CNN, in many other, that okay, finally a malaria vaccine has arrived. It was not actually a vaccine, it's just being to show that this vaccine can be made here, and we did some initial studies to show that this is able to create immune response. Then that's the time I said my lab had grown too big, and then I thought I cannot manage in a small lab, and I decided to shift to tuberculosis. Tuberculosis was a big thing at that time. It is caused by a pathogen known as Mycobacterium tuberculosis, and tuberculosis today, if you look at it, it's almost everywhere in the world except for some of the developed countries where you hardly have any TB cases. In the US, where I recently was there about 1 week ago, a few days ago I came back, and there are hardly any cases. You can count the number of cases of TB in the US, in Germany, others, but if in fact it's the largest killer of human lives. No other disease, even HIV, or even the COVID does not take as many lives as tuberculosis. It's primarily the disease of the poor. 1/4 of the world's population is infected with TB, and India, Indonesia, and all these countries account for 60%, of which 26% of the TB cases are in India. So India has a unique distinction of harboring the largest number of TB cases in the world. People infected with TB have a 5 to 10% lifetime risk of falling ill with active TB. It's unlikely that you will get active TB, but there may be, it depends. One group of thought, a very small group, believes that there is nothing like latent TB, or body clears it. But one group, majority believes that there is a latent TB, and you have a risk of 5 to 10% of getting an active TB. So what is this conversion from latent to active TB? We have absolutely no idea, and that's something we are working on. Success rate for curing for multiple drug-resistant TB. Today there are MDR TB, and because TB is a poor man's disease, hardly any investment from industries and big companies. So there's no return on investment. So there is no new drug. The last drug that was developed was 50 years ago, rifampicin. Till about 2 years ago, 2 new drugs have been fast-tracked, and delamanid and bedaquiline. And both these 2 drugs are also now, we are seeing resistance to even these 2 new drugs. So in fact, if some of us get, God forbid, these two drug resistant, there is no way you can be saved. You will still have to, the TB will take its toll. And most often, at least in countries like India and Africa, the improper use of antibiotics has been primarily responsible for, and also not continued use of antibiotics. It's a very slow growing mycobacteria. It divides once every 18 hours or once every day or so, as compared to ordinary bacteria that takes about just a few minutes. So it's a very slow growing. So the drugs also need to be given for 6 months. So 6 months for 4 drugs in a row, people don't take. They stop it after 1 or 2 months when they see that the drug is acting, and that's where you develop resistance. So this is a problem. In fact, when I was with my clinical colleague in Hyderabad, Dr. He is no more at Mahavir Hospital, Dr. Reddy, that is the time when Bill Clinton decided to come and see how Mahavir Hospital is achieving a success rate of 94% treatment of TB. I was invited to this function. I was sitting in the first row, and when I saw Bill Clinton, I said, why are people blaming Monica Lewinsky? She couldn't be blamed. This was such an attractive personality, neither girl would have done it. Anyway, so that's a different matter. So we looked at some proteins which were, initially we were looking at how the TB bacteria has been evolved. My group has been working over a long time on TB now, almost more than 2 or 3 decades now. And we published a review in the Nature Reviews where we showed that there is a reductive evolution, most unlike other places like Helicobacter pylori, where it evolves and acquires genes from outside. In Mycobacterium tuberculosis, the genes are lost, and there is a perpetual loss. Look at this graph here. It starts losing, and eventually this is the TB bacteria that you see here. And this is a member of Mycobacterium or some other pathogen, and over a period of time in evolution it loses. There is only one exception to this reductive evolution, and that exception is a gene known as PE and PE family of protein. P is proline, E is glutamic acid. So it has rich amino acids, proline and glutamic acid. That's why the name PEPP family of protein is known. It has 10% of the coding capacity of the total mycobacterial genome is dedicated to PEPP family of protein. Incidentally, this protein is not present anywhere in the living kingdom. You won't find it in plants, in viruses, in bacteria, in animals, nowhere else. In humans, it's not present. Why it is present only, and that to a very large number. And that's one of the things that we have been working on for the last few years. And we have been some very interesting leads that we have got, including this. And we believe that this protein is a ninja and also perhaps an Achilles heel. And more recently, we find that there are also, this protein may also be involved in conferring drug resistance. And that's based on recent studies that we carried out where we sequenced about 200 isolates from the northeastern part of the country and found mutations in the PEPP protein genes in drug resistant isolates. And that's something very, very interesting. I think Shekhar Raj did a good thing to me to give me the last slot, so that till coffee break I have time to speak. Right, Raj? You give that, that's the reason you give. I won't take that long, but I'll try to finish it. So there is a host pathogen interaction. Pathogen has its own strategies and host has its own strategies. Now this is where I think the interplay goes on. And there's a wonderful cartoon, wonderful real picture. Opportunist in taking support and yet no guilt. This fellow is sitting on a dog, happily sitting, so opportunist. But you know, totally, that's exactly what happens when you have a host pathogen interaction. They both really interact with each other, but they are not aware about what's happening, and they work only for, this fellow is working to sell his piece, and this fellow is happy because he'll get food after some time. So this is a kind of opportunity you have there. In the meanwhile, we also got access to a bacterium known as Mycobacterium W. This was a bacteria that was isolated and characterized by Prant Talwar when he was working at the All India Institute of Medical Sciences. When he moved as the director of National Institute of Immunology, we found this bacteria has, which was known as MW, has a lot of properties, including it was used to eradicate leprosy. And today, if we find leprosy is almost gone from India, the tribute should go to Prant Talwar for his Mycobacterium W. We thought, why don't we sequence this? It's a part of the Mycobacterium family. So we sequenced this Mycobacterium, and this was the first pathogen or a first bacterium sequence from India in event from India, and this paper appeared in a reasonably good journal. We have a lot of properties of this, and what we are now finding that this we named it as Mycobacterium indicus pranae. Indicus because it was isolated in India, pranae because Pran Talwar developed it, and NII because this work was characterized, and this was, as I said, Professor Anil Tyagi, Professor Akhilesh Tyagi, and myself. I was the coordinator of the sequencing project, and this is where we named it as MIP. And now MIP, we are finding, is able to give trained immunity. Trained immunity is a term that we immunologists use when we try to say that there is something in our body because of which new infections are able to be controlled, and that is what we find. This bacteria has properties to confer trained immunity. So to conclude about the work that we do in our lab, we do a lot of basic research, and we are very fortunate to get a very strong clinical partners. In fact, when I moved to IIT Delhi does not give lab to people who are superannuated. And like Professor Sopori, I also still work in my lab. So I chose to work at the Sharda University, which is in Noida, a good 45 kilometers, 1 hour journey from my home. But 3 days in a week I go. They have given me a hospital, access to hospital, and also to a cancer hospital, and it's not the occasion to talk about it. I'm coming here again for a TB meeting, I think in February 8, 9, 10, which is organized at this place here. And of course, we also do a lot of applied work at the IIT Delhi, where we have developed 2 new devices. CTB, we call it Chai Drishti. Chai Drishti, Chai is the Hindi of TB, and Drishti means to see. This is a very simple device that can look at TB bacilli by simply looking at ordinary microscope. So this has already been patented. This we have been the ICMR has taken interest in it. It has deputed this device into primary care center, so that we are able to detect. Our Honorable Prime Minister wants TB to be eliminated from India, not by 2035 but by 2025. It may not be possible because of the COVID problem, but nonetheless the problem is even one case of TB goes undetected, that person will infect 20 other cases. So it's very important not to leave any undetected TB on the place at all, and that's where we feel. And IMC square is immunomagnetic cell capture again. We are also working on repurposing. We have 4 drugs which are repurposed drugs. There is now a big project that has been given to us to do clinical studies on that. So that's where it is. There are several unanswered questions. New drugs against MDR, effective vaccine. There is no vaccine against MDR. There is only one vaccine, the BCG, which does not give lifelong protection because it does not give you what is known as a memory response. So we have some genes that we have been able to isolate, which gives a memory response. That's another project in our lab going on. Latency versus activation, how is it going on, and what happens, how does evolution impact clinical strain divergence? This is based on a study that we recently have been doing at the Northeast, where we have been sequencing more than 200 clinical isolates from Northeast. The synergism with other immunosuppressed conditions and impending quadruple trouble, TB, HIV, diabetes, COVID-19. In fact, COVID-19 was very interesting. We published a paper in Nature Medicine that has been cited, I think, 300 times already in less than a year, and that is to show how exactly this bacteria and double whammy of bacteria, TB and COVID could create serious problems. I was very intrigued with this quotation from Jacques Monod, "Chance favors a prepared mind." In the years that I have been working, I have been really trying to look at the chance to work on something very, very exciting. And I think we have been able to get some exciting things. This is a small nutshell of Professor Tom Blundell, Sir Tom, knighted by the Queen of England. He was visiting my lab, and we decided to take a photograph of the then lab team. We still have a very good group, big group that we have, and this is some of our group. Our group is actually a partnership between Dr. Weisberg, my group, and a group of Dr. Nasreen Etisham, who will be coming to wish Professor Sopori. She was working here in ICGEB, so wish Professor Sopori a happy birthday. So this is a small glimpse of the lab at that time. Now we have some of these people. In fact, I was in NIH to give a talk about a month ago. I gave a talk there, and there were 3 of my students who are right now at NIH. 2 people have just gone to NIH, and they were saying, "What is it that you do that they all go to NIH?" I said, "I don't do anything at all. They're good people." I've really been fortunate to get a very good people, very good young people who come and join us. and it's been a pleasure to work with young minds and learn from them. And I've been fortunate to get a large number of collaborators, some very top names. And funding support, people say if you superannuate you don't get funding. I beg to disagree. We still have a lot of very good funding from agencies, including big time funding from BIRAC, big time funding from ICMR, which is again till 2026, and these are really big time funding. So I don't think funding is a problem. If you have a good project, if you have a good science and good translation, of course there is a translational component, and if you have a good translational component, in fact Dr. Bhan, who is no more here, he was one of my real strong well-wisher and a mentor, I would say. When I was at the University of Hyderabad working for the university, the first time he gave a Center of Excellence grant, 23 crore rupees to us to set up a Center of Excellence. And after that, we did a lot of good work, at least we published a good and a lot of patents. And Center of Excellence went to second phase, and now of course they decided not to do, but second phase also we got about 13 crore of grant. So money is not a problem if you are able to do good science and do good. Thank you so much for inviting me, and I think I took 5 minutes more. Thank you. Can I use this water? Thank you.
Thank you, sir. I would like to invite Professor Sapori to give the token.
Those were times when we'll ask Professor Sapori to come and let's go and have a cup of tea. Those days, JNU used to be in the down campus, and we had wonderful times. Yes. Thank you so much, sir. Thank you, and wish you many, many happy returns of the day.
I've been keeping in touch and visiting his place when he was director, as vice chancellor, and IIT, who has been in contact, and also vice chancellor. He was twice vice chancellor.
But
I was with him that time.
It's been fortunate to have people like you to be with us. Thank you, sir. Thank you.
I was telling them we should call Naseem.
She's coming.
She's coming.
She is coming. She will come.
So this brings us to the end of the first session, and we have got coffee for all of you outside. Like Sir mentioned, we'll all meet on a cup of. But before we disperse, I must really compliment the two girls, because this is the first time we are running ahead of schedule, and we managed to save 10 minutes. So what we'll do is we'll have, can I just request all the speakers to please come in front. We'll have a group photograph, and then a group photograph with everybody. We'll just stand in up wherever, yes, and we get the photographer to climb up, and then we can just click here itself. And all the speakers can please, for this session, can please come in front. Thank you. next session, please.
Welcome back, everyone. Our first speaker in this session is Dr. Yashwanti Mutkil from University of Delhi. Her presentation is on DNA downstream and down the memory lane, journey starting from SKS lab.
ऊपर ठीक है. Good morning to all of you. It is very nice to see all of you on such a special occasion, and I'm going to talk about my journey in science, starting from Professor Sapori's lab. And very happy birthday to you, sir. DNA downstream and down the memory lane. So I will start with the DNA part first. DNA, I did my PhD on DNA topoisomerases, and I had privilege to work with Professor Sapori as my mentor. And actually I was registered under the guidance of Professor Sapori and Professor K.C. Upadhyay, but I did all my work with Professor Sapori, and Dr. M.K. Reddy was my kind of supervising mentor for the DNA topoisomerase project. And you could see the other mentor present during that time. They all had contributed a lot and provided me valuable suggestion, and I could successfully finish my PhD in 2002. Moved on to University of Toronto after that, joined Professor Daphne R. Goring's group, and she was working on the plant self-incompatibility, and I had opportunity to be part of a discovery of the novel E3 ubiquitin ligase family, which contain arm repeat proteins, and they are regulated by the receptor. They are major regulator in receptor kinase signaling in self-incompatibility. So the counterpart was discovered in Arabidopsis, and that's how I got introduced to the Arabidopsis system. Then I loved Canada, but it was very cold for me, so I moved on from there and moved to North Carolina, which is much better place in terms of weather. Joined Professor L.N.M. Jones' lab. He works on G protein signaling, and I guess this doesn't need any introduction, so I'm not going to get into the detail of G protein. You name any biological process and they are involved in it, right? So myriad array of functional kind of things, you name anything, you will find G protein connection there. So I was fascinated with the G beta mutant phenotype. You could see, which is shown here. It was previously established in their lab. It has enlarged root mass. I was fascinated. It's very easy system to score and kind of not very much time consuming. So I decided to work on the downstream effector of J beta signaling, which were majorly undiscovered at that time. So right here, I started my work from here. I used EASE-3 hybrid screen to find out the downstream effectors, and were able to successfully isolate a novel candidate previously discovered but not associated with the function. Arabidopsis and MYC downregulated like protein. They play a major role in terms of cell growth regulation, as they are along with AGB1, they regulate cell cycle in hormone dependent manner, and definitely they regulate, we established they regulate the PIN and the auxin carrier proteins, and that's how they carry out the function. And later on, G protein interactome initiative was also going on in the lab, and I was part of that. And after that, I moved on back to India and joined University of Delhi Botany department, and started my. So this, I'm not going to get into the too much technical detail of my work, as I have been told. So I'll just be giving you overview what I'm doing in my lab, what is the current theme and projects going on right now. So the overall theme of my lab is to understand how plants cell communicate through signal transduction pathway to regulate plant growth and development. And the main area which I am currently investigating is J beta gamma downstream effector. That's where the downstream part comes in. Downstream signaling pathways and cellular responses which regulate, and specifically I am focusing on the cell cycle, abiotic stresses and hormonal regulation of plant growth, and specifically the plant root growth fascinates me. So we are using multi-pronged approach, various kind of tools are employed. We further worked on the NDL1 AGB connection and were able to establish its specific hormonal involvement of this family member in hormonal responses as well as in salt stress, beta mediated salt stress. And I'm just going to take this effector of beta as an example and tell my story, okay? And there are, and keep in mind that there are a lot of beta effector like that, okay? So I'm just going to take this as a model example and tell what I'm doing. So we know that now we have established AGB and DL1 involved in various biological processes, and what are the downstream, further downstream components, and how the molecular mechanism is established. That's what is my goal to understand. So if I take NDL1 example and look back to the interactome, there are roughly 62 interactors could be found, and they all major 50% of them are involved in various kind of abiotic stresses, and some do play a role in the cell cycle and the plant growth also. So the way I said, I'm focusing on cell cycle and abiotic stresses. So a few candidates has been taken further for further analysis, and we have, so first you take those candidates to know the molecular mechanism, then you first of all you begin with kind of establishing their interaction in planta. Example is shown here for one of the candidates. And for NDL1, you can model, we are taking bioinformatic approach as well. You can take these effectors, you can model them, and then you can model like where they might be interacting on the surface of NDL1. You can find the interacting sites, and these can give you important clue how these protein might be regulated. And we are further proceeding with the biochemical characterization of NDL1 also, and that I'm not showing that story today, but we have made sufficient significant progress in that as well. So again, you can take functional genomics approach, that you can characterize these genes using knockout and overexpression phenotypes. Here the mutant phenotype of one of the candidate is shown, that it has root phenotypes, as well as stomatal phenotype is also there, indicating its involvement in the abiotic stresses. Further, the effect on the salt stress is also observed for this same interactor of NDL1, so indicating its involvement in the abiotic stresses. We are further characterizing this, but we are doing in vivo in plant experiment, but we have taken in vitro or the transient approach, and using the RD29 luck construct, we have established that NDL1 previously under non-stress condition is in cytoplasm, but upon salt stress, it binds to NDL1 and moves to nucleus, where this protein, VQ proteins are accessory factor of various transcription factor like Virkes. They can interact with them and then derive the expression of these salt tolerance or the other similar stress-induced genes. So this is about the project which was done by Arpana Katiyar. Most of it is, and Abhishek Kanojia is forwarding this work now. And another student is taking proteomics approach, and she tried finding the beta subunit mutant proteome under salt stress. We have identified few candidate proteins, and their validation has been done. And even further, their interactome has also been generated using in silico approaches. Both Nisha and Poonam were involved with this project. Sorry, what did I do? Okay, so then we are also under the abiotic stress category. We are also focusing on heavy metal stress, and Ayushi Jaiswal, as she is taking that approach, it was her idea. She mined the interactome and she decided to work on the the metal stress, and she could see a nice phenotype with both lead and cadmium, that this NDL1 AGB module, this negatively regulate the heavy metal stress response. and we are further working on that. Like, these are very starting points of that project. And so we started with what was known. We established various functions of this module, and then we were trying to find out what is downstream, right? So, so far we, as a part of DBT project, we started with a couple of candidates which were involved in root development, and we have characterized few to certain extent. The story is still kind of we are wrapping up those in the form of papers, and I'm not presenting today, but we started with 12 candidates, and for 2 we are about to publish, but we do have the interesting leads from the other NDL1 interacting proteins as well. So I wish to acknowledge my current lab members, which are shown in this picture, and this funding was provided by DBT, SERB, DST, and IOE DU also provides a small funding from IOE now. Okay, so now down the memory lane part of my talk. So this goes back to, I joined ICGEB in 1998. Actually, my coursework was done in the JNU, and after that you have to choose your supervisor. I was assigned Professor K.C. Upadhyay, but then during the coursework I had the opportunity to interact with Professor Sapori, and his teaching style really inspired me, and I got interested into the signaling very much, and I was after him. I used to go and approach him, can you take me, can you take me? And but he said, I'm leaving, I'm going to ICGEB. So then I requested Professor Upadhyay. He said, you can go with him, no problem. I'll be your co-PI and you can move. And sir asked me to write a small project. I wrote on artificial antibodies. He said, okay, write something. Then I don't know whether he remembers all this or not, but then I wrote a project and I kept kind of pursuing it, and then he said, okay, you can join my lab. And I seriously feel that was the turning point in my life, and it was a wonderful kind of decision on my part, or maybe divine intervention, I don't know. So when we joined ICGEB, they were starting up, and everything was new and empty. We had the whole long bench to ourselves, and nobody was there, and you can use almost whatever area you wish to use, and it has an open format, so you can go and interact with any group leader, and it was very, very open and inspiring environment. You can work day and night, and everybody will help you, okay? And you can see a lot of people from that time, Girdhar with lot of hairs, and Gayatri, Veena, Sir, Vinod Pant, and I think I don't remember his name, and yeah, the Jayant Chattopadhyay, right? Chatterjee. And again, I think all these photographs most of those are taken like on the same day. But this is much bigger group, almost entire PMP. Professor Sunil Mukherjee, very inspiring person, very much interested in science. You talk to him for a few minutes, he will reinstate your interest into the science if you're not interested for that moment. And Amos Gaikwad, Hoi, and Hop, and I guess Bien Singh, and I think I have covered pretty much everybody. Okay, so this picture is, this picture is during much later part of my stay. This was, I guess, my last day when I was leaving or something. So you can see the much younger generation have joined. You can see Snehlata, Richa, Mintu. Later on, ICGEB started its own PhD program, and the new candidates were taken up. So our juniors started coming up, and Dr. M.K. Reddy, and you can see Dr. Agarwal also, and what is her name? I'm sorry. Divya Rajagopalan, yes. And again, there was a cell signal transduction symposium, and Ralph O. Muller was part of it, and I think Stan Rooks was also there, the people who are part of today's event. And this is a poster session during that time. You could see Chandan, and Mamta Goswami was also there at that time. Okay, so with that, I will stop with a note which was written on, sir was correcting my thesis, and I don't know, I scribbled something and I left there because you are most frustrated at the time when you're writing your thesis, right? So I wrote Jeevan Kya Hai, and I just left it. It was on the last pages somewhere, and he write answer to that, and I wish to all kind of see that. Life is hope and an opportunity to realize self and his vast creation, to elevate yourself from the senses of individuality to universality, awareness of within and without as life. And this message stick to my mind, and I keep reminding myself this often, and I will stop with that. Thank you so much to all of you for your kind attention.
Thank you, ma'am. I would like to request Professor Sapori to give the token. Our next speaker for this session is Professor Girdhar Pandey from South Campus, University of Delhi, and he'll be presenting on exciting facets of calcium-mediated stress signaling in plants, how I landed into this field.
Very good morning to all of you, and I think we are extremely fortunate to have our supervisor, Professor Sapori. So I present a philosophical thought on how Professor Sapori actually have influenced life of many of his students, colleagues, associates. So I consider him as a shining star who actually lighting the path of many of us, actually many a galaxy of his students, associates and so on. So starting with my journey as a student in the actually SLS JU. So when I first interacted with him, I was so fascinated with the way he talked to me about the light signaling. I was very much interested in signaling and in the life of the plant. And he actually is kind of completely I would say brainwashed me to join him actually. So he assigned me these three major actually topics. One was the light signaling, because his lab was primarily doing light signaling mediated by cytochrome, trying to understand the nitrate reductase as a major. And there I think he asked me to actually look into the new facet, that is the isolation of the light mediated transcription factor by using the affinity matrix of the cis element and purifying these, because his laboratory was very well equipped with the protein purification, and so I think we tried to purify some of these transcription factors with the affinity matrix. And also I was involved with Dr. Meena Rani Chandok and Dr. Renu Deswal, who is also here, with purification of many of these protein kinases, stimulated by calcium, CAM, lipid. And then this was I think the major topic which actually is a part of my thesis. I think he actually was also working with Professor Alif Bhattacharya to understand these calcium regulated kinases, and Professor Bhattacharya's lab has isolated a novel calcium binding protein called, I think for the first time, and then he used this as a matrix, and we purified some of the protein that turned out to be the kinases, and I tried to functionally characterize it. So this is the way I say, like how I was fascinated with the signaling and how plant respond to the signal transduction, how plant actually responds to this stimuli, or how plant talk to the environment, how plant prepare themselves against any kind of adverse stimuli. So usually what happens is the cascade of the events start, that is the signal transduction pathways, and finally we see some kind of a response. So I don't want to go into the detail of this calcium signaling, but I think I should also mention, how this calcium signaling has actually taken place. What is the birth of this calcium signaling? It is so ubiquitously prevalent in all the organism, and it regulate a large number of processes, many of the processes, be it a stress condition or non-stress condition. So what happened is the prebiotic era, when there is a low concentration of the calcium in the environment, this primitive cell can easily grow, but as the calcium increases, because of the alkaline condition decline, and then the calcium increases in the cell, what happens is the calcium can easily chelate the phosphate, and then remove the energy currency. So cell has decided, the prebiotic cell has decided to remove this calcium from the intracellular environment and then develop the machinery, and that was the first, perhaps the development of the first kind of a machinery where the calcium homeostasis can be generated, where we talk about the involvement of the different channels and transporter. And then it's a wonderful, the life about the calcium, why calcium is only chosen as a signaling molecule, not the other element like magnesium, sodium, potassium. So this all depend upon the chemistry of the calcium actually. So when we talk about the calcium signaling, I think the major thing is the development of the calcium homeostasis. In all the organism, the amount of the calcium in the cellular environment, specifically in the cytosol, is very low, in 100 nanomole, and that is kept by the concerted action of the transporters and channels. So, and when there is a signal comes, the nanomolar concentration goes to the sub-micromolar concentration, and that is enough to trigger the downstream signaling pathway. And be it, some of these stimuli, adverse conditions are showing here, which can trigger the generation of the calcium, and then the calcium message can be decoded. Like many of these signal can be encrypted in the form of the calcium, and then the calcium is decrypted downstream, and then there is a response or adaptation is generated by the concerted action of many of these the machinery which comprise of the decoding part. So this is the typical way how the calcium signaling is shown, where there is a calcium signature is generated. This calcium signature is sensed by the calcium sensor, and then subsequently the downstream processes happen. And then in plant, actually the three major category of the calcium sensor has been identified. The typical one, which is a calmodulin-like protein, calcium dependent protein kinases, and then the calcineurin related system, which is called CBL and CIPK. So I just give you a brief glimpse, how I worked on this calcium binding protein from the Entamoeba histolytica and then the associated kinases. So in Professor Sapori's laboratory during my thesis program, so we basically functionally characterize it. When we overexpress this in the tobacco, we could see that can generate salt tolerance. And when this was, that time RNAi was not discovered. So people, most of the biologists use antisense approach, antisense technology. So we also cloned this in the antisense, and then we could see that it could give a very beautiful phenotype where the plant can, show alter morphology, and a high amount of the chlorophyll was detected in this. And we also isolated the protein kinase. Like I think this is the major, work of the Renu Deshwal. So where we could see this kinase was actually, actually binding to this sensor. We didn't know that was a sensor that time, but I think this might act as a sensor, and then this kinase was a calcium stimulated kinase. So moving forward, to my postdoc, I think I consider myself fortunate that I started to work on the field I like, the signal transduction. And then it is calcium signaling, and then I happen to be placed in a laboratory which is doing calcium signaling for postdoc, Professor Sheng Luan's laboratory in University of California, Berkeley. So who was starting the pioneering work of this calcineurin related protein, function of this calcineurin related protein. So CBL and CIPK has been identified. This is a calcineurin system in animal system, which comprise of the phosphatase and the calcium sensor CNB. CN is a phosphatase and also require CAM, and this can be activated phosphatase. This is inactive phosphatase, this is active phosphatase, is regulate. If you type calcineurin, you will see thousands of reports which are regulated by calcineurin. So what is happening in the plant system is, there is a total, I think the reminiscence of this calcineurin pathway has been found out. The calcineurin is absent in plant. There is the similar molecule called CBL, which is the homologs of the CNB, and it talks to the kinase instead of the phosphatase. So there is a paradigm shift here, but the analogy or functional analogy of this calcineurin related pathway is there. In Arabidopsis, there are tens of CBL, 26 of the CIPK. If you can see, they form the CBL and CIPK. Actually, this target the CIPK kinase, as I mentioned. So this can form a module, and this can actually regulate a large number of signaling pathway, which is shown here. So our goal was to understand the whole, many of these CBLs and CIPK, and try to find out the target of these module. So we started working systematically during in the laboratory of Professor Channel 1, and we found out that many of the CBLs are, despite of the fact they are very similar, they are actually regulating different kind of a pathway. We found out many kinases which are actually the target of this CIPK, and one of the target is the CBL9, target the CIPK3. And then many of these CBL and CIPK target the transcription factor like ABI5 and ABR that has been identified. Now this is the way how we say that many of the abiotic stresses conditions have been identified which are regulated by CBL CIPK. Similarly, there are different kind of a condition. Plant require a very high amount of the potassium contrary to the sodium in the soil. So this is, if there is a deficiency of potassium in the soil, which is supplemented by the farmers in the form of NPK, huge amount of the NPK fertilizer is added. So when most of the soils when are depleted in the potassium, the agricultural loss is happening. So this pathway has been identified, and number of these reports suggest that there is a pathway comprised of the CIPK 23 and CIPK 9, which regulate these the transporters and channels sitting on the membrane, and then regulate the uptake and homeostasis of this particular ion. So similarly, there is another kind of a kinase has been characterized, under the osmotic stress condition. This is its characterization, and then this is the typical pathway. CBL and CIPK is also a very typical kind of a pathway which was parallelly identified by Professor Jiankang Zhu's laboratory, which is known as a SOS1, SOS2 and SOS3. SOS3 is actually the CBL4, SOS2 is a CIPK24 kinase, and SOS1 is a sodium proton antiporter. So this is the typical pathway which actually was discovered by the Professor Jiankang Zhu, and in our laboratory we discovered that there are alternate pathway which are also present in the this is actually doing the efflux of the salt out of the cell. This is storing most of the salt or pumping the salt into the vacuole. That's why decreasing the cytotoxicity of the sodium. So despite of that, when we started to work on another kinase, this CIPK9 kinase, which is showing us which is being a critical regulator of the potassium nutrition, it also regulates the oxidative stress phenotype, and we found that this is regulating the oxidative stress, and this is the first time we show that it can talk to a mitochondrial gatekeeper called voltage dependent anion channel and regulate the oxidative stress tolerance. So this is the way how I think this is summarized. Parallelly, I think what we are now, what we are working on, another CIPK which is also, now this CIPK positively regulate the VDAC. Actually, this is the VDAC, which is a gatekeeper of the mitochondria, regulating the ROS homeostasis. And there is another kinase called CIPK3 which negatively regulate the VDAC. So there could be a fine tuning of this, actually the transporter or the channel sitting on the mitochondria by these two kinases and regulating the ROS homeostasis. As I mentioned, our laboratory is majorly working on abiotic stresses, but we are also, I think, finding interesting result when we are trying to understand the plant pathogen interaction. So we are also getting the biotic story developing with this. Since we have the tools of these CBL, CIPK channels, transcription factor and other component of the signaling pathway, we are trying to investigate into the biotic stress, and we are getting encouraging result that this one of the kinase called CIPK21, which shows a positive, which is positively regulating the osmotic stress responses, is negatively regulating the biotic stress. So that is the thing, very interesting thing, how these different stresses are cross-talking with each other. The biotic stress and then the abiotic stress, biotic stress and abiotic stress. In this case, one kinase is a positive regulator of the abiotic stress, and then at the same time it is a negative regulator of the biotic stress. So this is the story, the similar story, this module is also regulating the biotic stress positively. This is the data. This is also the story we are trying to develop. So based on this, all these data in Arabidopsis, the understanding of the working of the CBL and CIPK module, we have developed this model that many of the CBL, I'm just focusing on some of the CBL and CIPK, typically 23-9, which are majorly regulating the potassium uptake and homeostasis here, also regulating the osmotic oxidative stress. And then there are other kinases which are, this kinase is regulating the osmotic stress as well as the biotic stress negatively. And now you have another CIPK which is regulating the ABA and cold responses, and it is negatively regulating the oxidative stress by talking to the voltage dependent anion channel. So based on this knowledge, now I think the important goal of the plant biologist is to translate whatever knowledge we have in Arabidopsis or model plant, any model organism, can we translate, can we take it to the crop plant, can we develop the crop plant which can be basically tolerant to these kind of stresses. So using these knowledge, the component of the signaling pathways, which we say is a sensor, effector and a target, can we engineer these kind of a pathway in the crop? Because now, based on many of the reports, some of the report from our lab and other labs, it has been already found out that these pathways are highly conserved in dicot versus monocot. So whatever result we are getting, and genomically we are also, functionally also, we found that there is a high level of analogy. So what we are trying to do is we are trying to use two approaches using the CRISPR-Cas genome editing, where we are trying to target the negative regulator to make the knockout. At the same time, gain of function by, since this is a kinase which is actually phosphorylating the target, right? So now what happens is, if there is a phosphorylation on a target, this can be actually mimicked by mutagenesis, say for serine or threonine, it's a serine threonine kinase. This, if it is phosphorylating the serine or threonine residue, if there is a phosphorylation, there is a change in the conformation. That's how the effect happens. So if you mutagenize the serine or threonine into aspartate, this actually solves the same purpose as a phosphorylation switch. So what we call as a phosphomimic mutant. So we intend to develop, or we are in the process of developing the precise gene editing where we can convert the serine or threonine into aspartate and actually impart the gain of function. So this is the I think perhaps the last slide where we saw that this is the way we aim to develop the stress tolerant. The plants are growing under normal condition, under stress condition they compromise, the growth and development is compromised. If we can work with this negative and positive regulator in a genome editing approach, we can develop the stress tolerance. So this is I think a large number of a list of the people I am indebted, and especially Professor Sapori, my former supervisor, who actually inspire. I think I keep taking inspiration, and I am always motivated. As I mentioned, he keeps shining, keep shining the light of n number of the people, including me. So, and then I am also indebted to Professor Alok Bhattacharya, Professor Kailash Upadhyay, Renu Deswal, Meena Rani Chandok, Dr. Shiva Reddy, M.K. Reddy, and Amita Pandey, who is my wife also. She was also, the part of some of the story we have. And in UC Berkeley, we have a big group, the key people who were there, my former supervisor, Shen Wan, and then the Yongha, Bamgi, Kyongnam Kim, Sangchul, with whom I still collaborate. And in Germany, Münster, University of Münster, I still collaborate with Jörg Kudla and his group. And these are the past students, who has contributed immensely to many of the stories we have come up. And these are the present story. And then this is the recent picture, and then picture showing the Professor Christine Foyer, who is Bajra faculty in my lab, and then the adjunct professor with whom I am collaborating on this ROS and then the oxidative homeostasis management. Thank you very much for your attention. And at the end, or not at the end, but I think I should say first, without the financial, nothing can be done. So the financial institution who support the research in the laboratory are greatly appreciated, greatly acknowledged. Thank you very much for your attention.
Thank you, sir. I would like to request Professor Sapori to give the token. Our next speaker is Professor Renu Deswal from North Campus, University of Delhi. She'll be presenting on journey from GNU to Delhi University.
It's going to be a potpourri presentation with my personal and professional just glimpses of that, because the time was short, 10 minutes, so it's not possible to cover everything, whatever we are doing. So I did my PhD under supervision of Professor Sapori from 1989 to 94. Actually, I finished in 93 and I got awarded in 94. This is the time when Giridhar joined the lab. We overlapped, in fact in 93 I think, and then we did some combined work. And then I was in CPMB and NCPGR from 95 to 2001. In between 1 year of RA-ship I did. And in 2001 I joined Department of Botany as a reader, and I'm continuing there. So I would just like to give you some reflections of those times, and I really miss those times. Very exciting times, labs were open, interaction was open, and I don't find that now. There's something which is missing, that atmosphere, that very scintillating atmosphere. And in fact, when I joined for PhD, Professor Sapori was on, most of the time he was in fellowship. It was his peak time. He was on Humboldt, he was on Fulbright, and no senior student was there. The problem which I assigned, Glyxolase 1, to purify that from Brassica junctia, no senior was there, but that kind of proved a boon to me, because I had to learn things from different labs. And as I mentioned, this was one plus of SLS JNU. That labs were open. You can go into any lab, you can ask anyone to help, and they really used to help. So I learned everything from other labs. So the PhD problem which was assigned, as I mentioned, was to purify Gly1. And in fact, the stage was set already for that, because when I joined, it was a joint lab with Professor Shipra Mukherjee. And some of my older colleagues, they have already, Gly1 at that time, Glyxolase 1 was only known in animal systems, in human and animal system. Plants, there was no clue, but some indications were there. Professor Mukherjee, she did some work, and today only I came to know from Meera ma'am that Sumita is sitting here, they for the first time did some experiments to show that this kind of protein is also existing in plants. So I started with that, and as I mentioned, the column chromatography, and I'm a protein person, sorry, because I somehow mol bio never attracted me, because protein you can see results then and there. So I feel more clear, more satisfied working with protein. I could never imagine that in a test tube you are doing everything, and then I'm not a critic or something, but somehow it's my personal likings that I like to work with proteins. And eventually, at that time I was working with proteins individually, but later on I converted to proteomics, and I started using proteomics tools to answer some biological questions. So that is where I am now. I just pick up some interesting queries or questions, and I try to answer them through proteomics tool. So as I was mentioning, column chromatography, gel running, I learned from Professor Ashish Datta's lab. Natarajan and Anasuya, they were there, they taught me that. Then I had to raise antibodies, polyclonal antibodies. And for that, Apurva Sareen in Professor Rajiv Saxena's lab, she helped me. In fact, once I went to AIMS also, Dr. Bhan's lab, just to understand how to raise antibodies and just to see how do they do it. And that is how, it was a tough time. Another reason for being a tough time was my second PhD, my son was with me. I was married, I had him when I joined for PhD. So it was tough that way also. And another thing I would like to share, probably Professor Puri would also not register that or not know that, I'll just help him to recall. I was assigned a tissue culture project when I joined. Probably he did not believe my capacities or that I can work, because I had a kid with me. So, but I got bored of that. In 6 months I said, please change my problem, I cannot work on this, because it was very monotonous. Just transferring those, again I am not criticizing people who are doing tissue culture, because there are wonderful applications of tissue culture also, but somehow I didn't like that. So I told him, please give me something else to work on. And then Gly1, purification, again some seniors have already tried and they were not successful, so that was given to me, and that is where my journey started. And after that I joined, as I mentioned, my PhD was finished by 94, and these are the people who I would like to acknowledge today, on this day. Foremost, my supervisor, because signaling is one thing which connects all of us. That word clicks somehow, every one of us, because he was so interested always in that, and he explained in such a nice manner that we could never forget, and we cannot never forget that word. So somehow it comes into our work. And Professor Aksit Datta, Professor Shipra, unfortunately she is not there today. My supervisor, Professor Sapori, and Professor Upadhyay. These are the founder faculty members who founded CPMB, and I was fortunate to join there as a research scientist after my PhD. And then if you see on this one, this is the picture of some time when our labs were combined with Professor Shipra. You can see Professor Sapori, Professor Meena Sapori, then Professor Shipra, this is Meena who was working on kinases. Then some Kuljit, she was a postdoctoral person in the lab. Anita, she is in California now, I think. Vibha, she was Professor Shipra's student, and she was also there at that time when I was in the lab. So this is some gathering probably. And this is just a memory from CPMB, which has converted to NCPGEB at that time, National Center for Plant and Genome Research. And this was the first workshop which we organized, and some participants, again you can see some people which are still continuing in NFPGR. Praveen is there, Shubhra is there, she is the director now. So these old times pictures somehow I could dig out from collection, and this is what I did between in my PhD and postdoctoral stint, you can say. Gly1, that was my first love, so I somehow wanted to wrap it respectively. So some things we were doing, as I mentioned, we raised antibodies and we had purified the protein. So we used these antibodies to get some immunomodulation targets, because one of the property of Gly1 is that it was associated with proliferation, so that its inhibitor, they act as anti-cancer agents. So idea was, because we had antibodies, so we thought we will, I got a chance to visit this German Center for Biotechnology, or a fellowship, in Germany. I think I should go back. Can you just, I have to just go back to the slide. Okay, okay, thank you. So we did the peptide library preparation with the help of Professor Ronald Frank, and then I used those antibodies to know the immunodominant epitopes, because we wanted to kind of provide a strategy for immunomodulation of as a device, as Dr. Hasnan was mentioning about anti-malarials using epitopes. He has actually devised a vaccine for that, a probable vaccine. So it was on the same lines that we wanted to devise a strategy for anti-tumor device or anti-tumor thing, and this work was published then in 2005 in Peptides. Besides that, I think Girdhar has made my job easy, because that was another thing which I did in between. Another thing was, we showed for the first time that glyxase-1 is a calmodulin binding and calmodulin stimulated protein, and the interesting point here was that even magnesium was required for binding. We just know that calcium is essential for binding of CAM to proteins, but we could show that even magnesium is important, and that binding was dependent also on magnesium. And then as Girdhar mentioned, I think I'll skip this, we purified a novel kinase, and thanks to Meena, because she was the one who pioneered this kinase work, and we actually learned from her. And Viradhar, as he joined almost the same time, so then we did some joint work, and as Girdhar mentioned, we could publish some of this. So this was my postdoctoral stint. After that, this one more important point. I joined this EMBO and FEBS advanced course in Debrecen, Hungary. And this was a kind of turning point for me, because this is where I was introduced to mass spec machines. And at those times, mass spec machines they were occupying a room. One machine in itself was occupying a room. That was the type of mass spec I saw. And I was somehow got excited that we should work on this and we should work in this area. And by that time I also joined NCPGR, so there also that was the proposal that I will start work on proteomics. That is how I started thought of using proteomics as a tool to understand or to answer some biological questions. And then after this I joined botany department DU. And JNU is inherent, I cannot forget it. I could somehow never connect to DU. I am there since last 20 years, but that connection is not there. JNU is in my blood, I will say. I cannot forget JNU, it automatically comes. So we started work on nitric oxide signaling in plants. And that was again a new area at that time. Not many work was available, especially on, we were working on a post-translation modification called nitrosilation. Not much was available in literature, but as you know, some places are, there is a lot of inertia, so you need some time to develop your facilities. And so by the time we published our first paper, before that another paper in plant phase on nitrosilation appeared from Arabidopsis. But anyway, we could contribute this beautiful work from nitrosilation, where we observed that rubisco is coming as a target of nitrosilation. That was an interesting observation. And in brassica we could show that this information was already there, that cold stress leads to kind of inhibition or down-regulating activity of rubisco. In fact, many stresses, they lead to down-regulation of activity of rubisco. So this was one question, and nitrosilation actually could explain, nitrosilation of rubisco could explain almost 40% inhibition of rubisco during cold. And this paper was published in proteomics, and it is a highly cited paper of that year and later on also. And this continued, and this is another interesting findings from nitrosilation work, where we could show that glycolysis is tightly controlled by nitrosilation. And we could also see PTM crosstalk between all different NO PTMs. I will quickly go through my slides, just 2-3 slides. Then another piece of interesting work which we are doing is on antifreeze proteins from a Himalayan shrub called sea buckthorn. This is the berry of the shrub. This is how the shrub stays in Himalayas. And so we have been able to do lab scale purifications, and also been shown using one purified AFP for cryopreservation of RBC. So we are here with this problem, and we want to launch from here because we have all the tools, so we want to go for applications of this. And this is another work on cuticle proteome of brassica, where we have used different brassica varieties to get, again, the point here was cuticle proteins, very lesser known, very few reports, just one report from tomato fruit was there on cuticle proteome when we started this work. And we are getting some interesting leads. We have seen myrosinase on surface, similarly antifreeze proteins or antifreeze activity on surface, which is not reported yet. And this myrosinase generally is a protein which is sitting inside the myrosin cell in the cell, and it is giving glucosinolates. It's working on glucosinolates, and those are again anti-cancer things. So we are continuing this myrosinase story, and also this AFP work we have recently submitted. And this is another interesting outcome of the cuticle proteomics work, where we have made cutinsomes, which are nanoparticles containing cutinsomes actually transfer LTVs to cuticle. They help in formation of cuticle structure. So, and this can also be by certain chemical reactions, you can convert this into sheets. So, the idea was to create bioplastics, and the major idea was to translate this to rice, because we are having a constant problem in northern India in winters of that parali burning and all. So, how to use this agro waste to bioplastics, and then it can be utilized. So, we are again, proof of concept thing is there, we have to go further with this. And this is another thing which is keeping us busy right now. We are working on NP protein corona. NP is nanoparticles. Nowadays, nanoparticles are being used by many groups without bothering about the safety concerns of NP and without knowing how what they are doing in the biological system. So this is one question we are asking ourselves: what happens to a nanoparticle once it enters the biological system? Definitely there are a lot of interactions with different biomolecules, and proteins are the major one. These reports are already there in animal system that proteins is a major category of biomolecules which interact with NPs. And the fate of this NP, its reaction would depend on these proteins which are binding on this NP. So we are trying to decipher that again using proteomics approach, which kind of protein are binding, then we are comparing different NPs, because this will depend on different types of NP and different types of plant system, because the protein origin from there. So we are trying to, we have initiated this work again. This is something NP protein corona work is just kind of, it's just beginning, it's just starting. So there are again not many information on this in literature. So but again, this is a very exciting area of work in which we are engaged, and what we have shown that we have compared the extracts from nuclear and protein extract, and we have shown differential binding. That is what very obvious, that is very natural, that you will have different type of protein binding. But what is the benefit of doing this, all this? Because you can then design your nanoparticles and you can select the applications. If you can somehow know the procedures, the molecular mechanism, then you can do an informed designing of NPs, and then you can use them for various purposes. So right now, as I mentioned, not many people are there who is working on that area, so little difficult but interesting and exciting. And this is, we have published some special volumes on proteomics, and we also conduct workshops on proteomics because I believe in sharing. So whatever we have learned hard way, I want to share it with everyone else. So we are having, every alternate year we are having plant proteomics workshops since 2013. This is, we will have one this year, and we are training college teachers, research scholars and teachers from all over India, and we have a good representation here and many people attend this, and we have, I'm happy to share, we have trained almost more than 200 people in these workshops, and that is satisfying. And this is the group at that moment. It has now turned over and some new people are there, and whatever I presented is marked in this people, student who has contributed are marked in red color. Their work I have shared with you. Rest, a lot of different problems, and I'll say I'm a very effervescent person. I choose problem according to my likings. I don't bother about anything else. So that is a problem with me, and that's why you won't see many continuation or things like that. So, and I think the last slide. Thank you for your kind attention, and Jai Hind. Thank you.
Thank you, ma'am. I would like to request Professor Supori to give the token.
Just one last comment. I'll use the mic. There are few properties or few characteristics of Professor Supori which I cannot never copy. He is self-disciplined. He is there in the lab by 9. He is following the literature, the way he follows, and his interest in science. I cannot match that. I cannot match that, sir. And this all have, whatever little we could do, it's all because of these characters of yours that inspired us a lot, and we are still trying to do something. That is all I can say.
Our next speaker is Professor Raghuram from Guru Gobind Singh Indraprastha University, and he will be presenting from his topic is From Plant Signaling to Sustainability, a journey from PRL to JNU.
Thank you. Happy birthday, Professor Sopori, and thank you, Niti, Rajsekar, Arun, all the people who have worked tirelessly to make this event possible. For a generation which was trained to specialize, focus, narrow down, I probably took the opposite track. I kept expanding, went out of focus so much so that it appeared that I am a man without focus. But I'll try to show you that there is a method in the madness, and one discovers that along the way. Well, a similar photograph was shown by Renu about the PRL group. Must have been around 89, 90. Renu and me also share many things, our strong likes and dislikes, and also our birth date. We were born on the same day, same year, and she was also my batchmate in PhD. And both of us pursued our own passions as we went along. This was me, just to prove that I was also working. Because most people thought that I was not working. And I was not considered to be a guy who was even, having any potential to work in the lab. That's because most of my work used to get done in the nights when there was nobody else. And Professor Sopori never came at 9, he came at 8. And I knew it because I worked the whole night and I used to go out and he would come in. And we often used to communicate through chits because I was hardly there in the day. Of course, another thing that I must acknowledge, most of you know it, is that JNU is a very interesting place, a very different place where people do different things. So the first thing I did differently in JNU was to become an activist. Much to the irritation of Professor Sopori, there were a few others, I think probably in Professor Sipra Mukherjee's lab, Atanu was there, Astor Doshi had also been having some brush with activism for some time. He died an untimely death. But Professor Sopori's lab was a very, saintly place. So I was somewhat of an unlikely person to be in that lab. Though most of my activism only focused on misconduct, either scientific misconduct or financial misconduct. I never did any other activism, at least in those days. But it was probably thought that, many people thought that I might have a better career in politics. But I also did some work in the middle of all that. So the second thing I did differently was I found my love and got married in, I think, the second or third year of my PhD. So this was Professor Sopori when he visited my married student's flat in Mahanadi with Meena ma'am. This is my wife Madhvi, who's a scientist in NISTAD's CSIR, which is now renamed as NISPR, National Institute of Science Policy and Communication. Anyway, now to some science. Molecular biology was my calling. That was one thing we differ. Renu worked with proteins, I worked with transcripts, and I still work with transcripts. I had some bit of an advantage as an MSc in life sciences with more integrated approach. At a time when botany zoology were the right thing to do, I actually fought against that and went life sciences. And I had an opportunity to, in the first batch of the UGC JRF, I did MPhil in zoology in Pune University, where I probably got the first MPhil degree in genetic engineering in those days. Cloning bacterial cellulose genes and expressing them in E. coli. So it gave me some grounding in molecular biology techniques and approaches. and also it further strengthened my interest to do molecular biology. I took some time off. After that, I thought I knew enough about biotechnology and molecular biology to now directly go and apply it in the industry. Went and joined Thapar Group in Patiala, where I was, before the term biofuel was coined, we were actually producing biofuel, bioethanol, using molasses in immobilized yeast cells, where I isolated the strain, standardized the immobilization conditions, and we could produce ethanol continuously at 9% volume for up to 6 months in an inclined column bioreactor. That technology was eventually transferred to Bharat Starch, and we had a couple of modest papers and one patent application that came out of it. But then I soon realized that industrial R&D can get very boring. So after a year I left and came to JNU. I had to qualify NET again, take all the exams again. Initially I was assigned Professor Kale's lab for radiation biology. After the coursework and some period of working in his lab, I was not finding enough molecular biology in that lab, and I still was very fascinated to do molecular biology. Professor Sopori had just returned after his Humboldt Fellowship in Germany, and the PRL at that time had already transitioned from plant physiology, tissue culture to plant biochemistry, and was actually on the throes of transition towards molecular biology. So when I came and requested him for a change of guide to join Professor Sopori, he said, if you can do this, you're welcome. If you can put your training to work in my lab, you're welcome. So I changed the lab, but because I came from an earlier background in cancer biology, and I also did a term paper on tissue-specific expression of oncogenes as a part of my MPhil coursework. Cancer is a disease of signal transduction. So a lot of my interest in signal transduction, molecular biology, nexus came from cancer biology. So I initially started looking for oncogenes in plants, which Professor Sopori did not laugh at when I said, I want to, so he said, what do you want to work on? I said, I want to look for oncogenes in plants. So he didn't laugh at it. So I obtained probes from CCMB from various places, and we did southern blots, and we found some signals. And after that, for CMYC and human HARAS and a couple of other oncogenes, we found homologs in, at that point, sorghum, I think in maize we were working on. Both did some signals, but we did not really know how to move further after that. In the meantime, he got a CDNA probe for tobacco nitrate reductase. So he said, okay, we have now you know how to use probes and do southerns. You can do this for nitrate reductase probe, which Wilbur Campbell had provided us at that time. So also try on this. And then gradually I moved into nitrate reductase work, and for the next 5 years, my southerns and I think I even did some northerns on oncogenes in plants just remained buried in a pile of papers. It was in the 6th year of my PhD that I went back and recovered those southerns and northerns, because they were the only proof I had that a G protein existed in maize. Because a RAS probe, a small G protein probe, was still homologous to the heterotrimeric G protein in maize at that time, and it also produced a transcript of a size of a G alpha subunit. So I could bring back that G protein into the nitrogen story, and that's how the story moved. So another thing I did differently in PRL was, this used to be the usual scheme of theme around which all work in that lab used to revolve. By the time I finished my PhD, I pushed from nitrate as a side business to nitrate as the main signal for NR gene expression, and it is phytochrome which is the side stimulant. And that little change I did in this continues to be the main theme of our research in my lab till date. Now we work on nitrate response. I'm probably the only one among his students who still works on nitrate response and a bit of nitrate reductase and other genes involved in nitrogen response in plants. So this was a very early paper. In fact, I contributed, I think, the first transcript data to Arun Sharma's paper when he came back from US and restarted some of the unfinished work. The other thing I learned in Professor Sopori's lab is to make models. Always propose a conceptual scheme for what is happening. Interpret your data and try to build a story around it. Even if some of it is hypothetical, some of it is real, it forms a basis for what you do later. And it requires tremendous courage of conviction to propose a model. At that point, it was like an adventure for us. We did not understand how much conviction it required, but now we are on the other side of the table, so we know how difficult it is to put forth something like this and get away with it in a prominent journal in the field. We still do that. As and when possible, we come up with these things. I spent all my 5 years doing dot blots and northern blots, which have all disappeared now. They've all become extinct. So I thought it will be a bit of nostalgia to take a look at methylene blue stained northern blots and autoradograms and dot blots, which have all but disappeared. This 1995 paper still gets cited occasionally. Also probably the first paper in plant molecular biology from JNU. In one lab meeting, Professor Sopori said, plant molecular biology mein paper chhap ke dikhao. I said, yes sir. Everybody laughed. I was not considered to be somebody who could actually do that because I was never seen working. So I was always busy doing other things. But eventually we did. Luck was on our side and work was not bad. So we made it. In fact, just last month, our only second paper in plant molecular biology got published as a review on G protein signaling. So from 1995 to 2023, we came back full circle. So this was the story. Initially, we did the these retrieved blots, which showed that a G protein RAS homolog exists in maize genome. Then we did northern blots to show that this gene was transcriptionally active and its size corresponded to what would be G alpha subunit. Then with the help of Mina, I did some western blot to show that only light exposed membrane fraction bound radioactive GTP, whereas ethylated membrane fraction protein did not bind GTP. So we showed existence of the gene, we showed the transcript, and we showed that the protein was responding to GTP. So this got published after I finished my PhD and moved to Bombay University after a stint of a couple of years in journalism, scientific journalism, as a correspondent in Down to Earth, where I learned to look at science as if I don't belong to it. As a science journalist, you get a ringside view. But if you were only an insider just yesterday, so with the advantage of an insider of science as a PhD from JNU, you get the vantage point of looking at it as if you don't belong to it as a journalist. So this gave me a very interesting experience, and I put it to good use later. The journalistic skills, the writing skills, the editorial skills, and also the ability to simplify things, especially for policymakers, for non-specialists, which we continue to leverage even till date. So nothing ever goes waste. So that was probably the most out of focus thing I could ever do. After PhD in JNU in plant molecular biology, go and join a journalist. But I still think that also gave me a lot of things which I still use back again in molecular biology. Not many people were fortunate to actually come back from what was considered to be a one-way traffic. You move out of science, you never get back to science. But within 2 years, I was able to get my first job in Bombay University, and we did that. And then I spent some time trying to put this whole story together, and the original manuscript, when I finished my PhD, Professor Sopori wrote, needs more meat. I think that sheet is still somewhere with me. But then I could not produce that extra meat even after I went to Bombay. Things had changed. It took me a long time to set up my lab. So we wrote it up in whatever way we could and just got it published. But a few years later, once I had my lab and my student, we were able to replicate this work in rice, which became the model system for my lab, where we showed that cholotoxin was mimicking light effect and nitrate effect in inducing NR gene expression in dark adapted rice leaves. This was a decade before the first paper gave genetic evidence of involvement of G proteins in nitrogen use efficiency. This came in 2014. My work was probably done in 1990. We don't get cited, that's a different matter, but the fact is we were probably looking at the right thing at the right time. And then we did some, I had a fellowship to UK, in fact with Richard Hooley, whom I met here in this hall when Professor Sopori organized a plant signaling meeting. And then I spent some time with him, took his G protein mutants and looked at the transcription accumulation under different nitrogen doses in wild type G protein, G alpha mutant and GPCR mutant. We could see that some of the genes were underexpressed, both for nitrate lactase and ADP glucose pyrophosphorylase small subunit. Their expression was impacted by G alpha mutation, to a lesser extent also by GPCR mutation. So this gave me the confidence that I can still extend that story that I started in his lab. And then in a few years we published a lot of work, initially starting with Arabidopsis, but later also going into rice. And in the process we also resurrected GPCR, which was thought to be not required for plant G protein signaling, when Alan Jones and Sarah Assman had a dispute over some yeast to hybrid interaction studies, and finally that idea that GPCRs are not required dominated the entire literature. With only half a dozen groups doing G protein research in the world at that time, even to publish this work was very difficult. It took 2 years of struggle to publish our own papers with firm genetic evidence that no, GPCRs matter and they often regulate the same genes and same processes that G alpha regulates. May not be all of them, but some of them certainly do. So this is that paper I was talking about, Life, Death and Resurrection of GPCR, just appeared online a month ago. Then we moved into nitrate response. By then the whole story became much beyond nitrate reductase. Many more genes are nitrate responsive. If you want to improve nitrogen use efficiency, you have to not look at one gene but many genes. So we had to do microarrays, and we also found commonalities between G protein responsive genes and nitrate responsive genes, a story that continues till date. I'll move quickly on this nitrogen use efficiency story, but just to tell you that there are many steps where biological interventions of nitrate response in plants can be addressed, and rice is a very good model system because it takes away almost 1/3 of all the fertilizer that we use in India, goes only into rice and only rice, and 2/3 goes into rice and wheat put together. So these are the best crops if you want to improve nitrogen use efficiency. So we tried to, because there was no proper phenotype, we tried to develop the phenotype and then use that to characterize the genotype. So this was our paper where we showed screening 21 different varieties of rice across agroclimatic zones, and we found out in collaboration with Sitaramam that germination could be used as a simple phenotyping tool. Then we did a more detailed lifelong phenotyping study in the greenhouse. Out of 25 characters, 20 were nitrogen responsive, and 12 of them responded to yield, which is important for NUE, and only 8 of them directly associated with NUE. Then we did the microarrays and shortlisted the yield associated and phenotype associated genes through hierarchical shortlisting. We are now doing trying to do CRISPR knockouts for these candidate genes that we have shortlisted from 1,000. We also, because we got a Japonica mutant in G alpha, we were also parallelly working in Japonica. So we have both rice indica and Japonica going together, and we bring out with both. Then we identified contrasting genotypes for rice nitrogen use efficiency and showed that these are the processes in which the transcriptomes very clearly differ. Photosynthesis, cell division, transporters and transcription factors together account for most of the contrasting transcriptional patterns in two contrasting varieties of rice identified by nitrogen use efficiency. So this is further characterization of that. We later brought out a special issue of Frontiers, where we also now lead other work in the area by other people. Another special issue of Frontiers is right now open, if anybody is still interested. From there we move to a wider issue of what happens to nitrate when it is not taken up by the plant, it becomes a pollutant, and nitrogen is already crossed the planetary sustainability boundaries, and it comes not only from agriculture but also from livestock and traffic and waste management and power sector and industry. We then got into setting up the Indian Nitrogen Group. We got affiliated with the International Nitrogen Initiative. We did the Indian Nitrogen Assessment, where a lot of other people in environmental science and other sciences came together, and we showed that India is actually a hotspot for nitrogen pollution, for NOx, for nitrous oxide, which is the greenhouse gas, as well as for ammonia. And this is the declining nitrogen use efficiency with the increasing fertilizer use in the last few decades. This was a report we did for UNEP on the nutrient world. So the good thing is that we were early starters in recognizing the importance of nitrogen for sustainable development, almost at par with the rest of the world. So therefore, we were able to lead the INI, we were able to bring the UN resolution on sustainable nitrogen management through the Indian government. Now Indian government actually co-chairs the global intergovernmental cooperation for sustainable nitrogen management. The nitrogen working group of UNEP is now jointly chaired by India and Romania, and this was just last month's development. And it connects all the 17 sustainable development goals. Attaining all of them will not be possible without doing something about nitrogen in each of them. And these are some of the more recent environmental science work that we are parallelly doing in collaboration with other people. Just to tell you how and where science takes you, we are also looking at nutrient quality in wastewater in the sewage treatment plants of Delhi, and we hope to do this. Sometimes we have to also spend a lot of time generating policy reports for bureaucrats and governments and UN bodies, and that's our journey. This is where nitrogen is at the moment. There are a lot of international framework conventions and bodies which talk about nitrogen, and we give inputs to as many of them as we can. And these are various special issues of nitrogen. Now every year there are multiple special issues coming in multiple journals, and fortunately we are able to contribute to many of them. Last but not the least, I am proud to say that we were also able to build an Indian journal of Indian origin to an Impact Factor 3 journal last year, and this June we are hoping to make it Impact Factor 4. And this is the most affordable, almost free publication source for Indian plant biologists, and also for people across the world, because half the papers that we publish now are from outside India. Thank you very much.
Thank you, sir. I would like to request Professor Sapuri to give the token. Our next speaker is Professor Arun Sharma from South Campus, University of Delhi. He'll be presenting on role of glyoxylase enzyme in ripening of tomato fruits.
Yeah, first of all, I would like to wish Professor Puri a very happy birthday. I joined his lab in 1980, so like if I start counting, it's like about 22 years gone, sorry, 42. So basically, one thing I would like to say here, usually, when it came to having good luck in any sphere of life, I thought it was not on my side, but the best stroke of luck happened when I joined Professor Sapori's lab. I got the admission through waiting list in my batch, but I got the best lab despite of that. So that was the best stroke of luck for me, and which has helped me a lot. So for this event, I have chosen the topic which is very close to Professor Sapori. This is related to Glaxlase enzyme, and this is the work which we started very recently. In fact, in my early part of career, Professor Sapori gave me clones for Glaxlase 1 and Glaxlase 2 to try and see whether they have some role in tomato fruit ripening. I couldn't do work at that time for some reason, but recently we picked up this work and we tried to understand if Glaxlase has any role in tomato fruit ripening. And this is the area we have been pursuing, different aspects of tomato fruit ripening. So one thing I would like to point out about Glaxlase. See, this is the very important step of glycolysis, where dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. And during this process, you have a intermediate which can be converted to methyl glycol. Now, this reaction, which is kind of side reaction, is stopped by a loop. There is a loop in this enzyme, 10 amino acid, and this restricts this reaction. If you see the reaction in solution without this loop exerting its role, this side reaction is 100 times faster than the main reaction. So that means during this process, if this is not functional, you will produce very high amount of methyl glycol. And this is the toxic kind of molecule which affects so many molecules. It can, modified proteins, nucleic acid, and it can create a lot of havoc. So basically, this is one source of production of methyl glycol, and Glaxlase neutralizes this toxic molecule. So first of all, we tried to see during tomato fruit ripening how the level of Glaxlase changes, how the level of methyl glycol changes, and we checked that level of methyl glycol. So what we see that during early part of ripening, when fruits are green, amount of methyl glycol is higher, and as the fruits start ripening, this is the wild type, breaker stage, amount of methyl glycol decreases, and red ripe stage still decreases further. Now another interesting part is we also checked this level in some of the mutants which are mutants for fruit ripening. And one of the very important mutant is RIN. This is mutated in one of the Matz-Cox transcription factor, DMATS-RIN. And in this RIN mutant, most of the ripening related changes are compromised. Like tomato fruits don't develop red pigment, they don't soften, they stay very firm even at the mature stage of ripening. So many of the ripening related genes are knocked out if this is mutated. Now you see in this case, methyl glycol level is very high. So in green fruit it's high, and also this RIN mutant, they never ripen, they stay green or yellowish. In another mutant, NOD, which is also ripening mutant, which affects a lot of aspects of ripening, pleiotropic effects are there. Here also level of methyl glycol is very high. So when methyl glycol level is high, fruit ripening is not happening, it's compromised. So that's the take home lesson. Then we also checked if we treat fruits with methyl glycol, can we prevent ripening? And we tried treating fruits. Normal fruits would ripen, but once you treat with methyl glycol, you can see as you increase the concentration, it's not becoming red. So methyl glycol affects the ripening process. We also check activity of glycolase enzyme in different fruit ripening stages and also in the mutant. So in mature green you don't see much change, but you can see here in the mutants, ripening mutant, level of glycolase 1 activity is almost knocked out. Gly2 activity is also reduced, but not so much as Gly1. So in ripening mutant there is lack of glycolase 1 activity, and that's why probably there is increase in methylglycol, because it's not getting neutralized by glycolase. So to study further, we characterize glycolase gene family from tomato, and these are the genes which these are of different categories. Like there are some with 2 domains, some with 1 domain, and there are some of Gly1 category and then Gly2 category, and there is a Gly3 category, which is usually common pathway is that 2 enzymes are involved, 1 and 2, but in Gly3 this is a single enzyme which can catalyze the reaction. So then we checked which of these genes, Gly genes, are showing fruit specific expression, and we found that these are the 3 genes which were showing fruit specific expression. So we thought these could have role in tomato fruit ripening. Then we tried to silence these by using wicks, and there we found that this is the one which gave phenotype. So then we picked up this and used for further characterization. So if you see the sequence of promoter of glycolase, this is Gly14 gene. You find various elements which are specific to fruit development, fruit ripening. There are EREs, EBOX, there is ethylene biosynthetic element which is there in ACS2. This also has CARG box, and I told about the expression in RIN mutant. In RIN mutant, expression is compromised. Now this CARG box is the binding site for RIN transcription factor, and which is also here. So then we tried to see whether RIN regulates Gly1 activity. So we used reporter assay where we had GFP fused to Gly, and then we add another effector molecule which is the RIN, and then we see the fluorescence, and this data indicates that RIN binds to Gly promoter and increases the expression of its promoter. Now there are also these ethylene responsive elements here, so it's likely that it will respond to ethylene, and we tried using ethylene. So if you use ethylene treatment, then activity of Gly goes up, and as a control, this is the E8 gene, which is very well characterized for ethylene response. So we compared the response with this, so they got similar response. And then we should try to inhibit ethylene by using inhibitor. Then this is negated, ethylene induced increase. So this shows that glycolase gene responds to ethylene. It has ethylene responsive element. And then this binding of RIN we characterize by mobility shift experiment also. So we express RIN in bacterial system and use this RIN for mobility shift, and this showed that RIN can bind to Gly promoter. We also characterize this interaction of RIN and Gly promoter by this yeast to hybrid acid. Then we tried to see if we knock down the activity of glycolase, then what would happen. So we use microRNA based silencing vectors, and then try to silence the activity of Gly14. So these are the 2 lines using microRNA based vector where ripening specific promoter has been used. This RIP is the promoter, ripening specific promoter, which we identified in our lab and characterized. So these lines were made using fruit specific promoter, And then we also made lines with 35 promoter. And as you can see in this case, this is the normal ripening where you see red pigment. If we knock out GLY1 by using fruit specific promoter, knock down, sorry, you can see that this pigment is not coming. These are two lines. If we use 35 promoter, then in addition to this block of ripening, there is also developmental defect and size is very small. So that shows its role in early part of development also. In this case, since the promoter was ripening specific, fruit could develop to the normal size and then later part of ripening was affected. So we also checked in these lines whether GLY1 activity was really inhibited, and we could measure that whatever we tried, we got the inhibition of GLY activity. Now what happens when GLY activity is reduced, methyl glycol level increases, and one of the property of methyl glycol is it can glycate proteins. So when we knock down GLY, we expect Mg to go up. So these are the glycation product. So this is in mature green, this is in the wild type, mature green, breaker plus slow, breaker plus 6, and then these are the lines where GLY has been silenced by microRNA based silencing. And you can see the signal, you can compare this with this, this with this. So there is very high amount of increase in glycation product. So if you silence glycolase gene, then there is increase in methyl glycol, and this methyl glycol then modifies these proteins which can be seen, and via this interaction at the protein level, methyl glycol can affect the activity of several genes. Okay, so next question was how does GLY4 affect fruit ripening? And so if we see the ethylene level, this is wild type, and when we silence glycolase, you can see ethylene level is going down. So we know that ethylene, but then how does glycolase affect ethylene? Which step might be the target of glycolase? So we try to see when we see this inhibition of ethylene and inhibition of ripening, if we give external ethylene, can we compensate for the loss? And here you see that these lines, when we provide ethylene, there is ripening. So that means ethylene synthesis is compromised. So then which step in ethylene synthesis might be compromised? We try to identify, because methionine is the precursor for SAM and then ultimately ethylene. So we check the level of methionine, also the amino acids of methionine pathway, and we found there is some decrease in methionine. So we try to complement by using methionine, but methionine could not complement very much. But then we tried ACC. In this case, this is the complementation with ACC, and here you can see that if we provide ACC, then this block, whatever glycolase cause, can be overcome, and we see ripening. You can see in this figure, when we give methionine, there is no complementation in the glycolase silence line, but when we give ACC, you can see red pigment coming up. So that means somewhere before ACC is generated, there is an effect of glycolase. So this is the model we suggest that when fruits are green, Mg level is high, and during ripening, glycolase is induced. There is a ripening specific glycolase which is induced, that brings down the Mg level and leads to ripening, and bringing down Mg level leads to this, this is activated and ethylene is produced. And when RIN is mutant, then RIN affects the expression of this gene, glycolase is down, and that's why you have more of methyl glycol accumulated, and that blocks the ripening. And so possible somewhere here where you have SAM generation, and from SAM you have ACC produced, somewhere that one of these steps is blocked by the action of methylglax. So this is the role of glaxlase that we found in case of tomato fruit ripening. It needs more work, and I think I'll stop here, and thank you very much for your attention.
Thank you, sir. I would like to request Professor Sapuri to give the token. Our final speaker for this session is Professor R.P. Sharma from University of Hyderabad, who will be presenting on Seeing the Unseen, a trifoliate mutant allele fortifies folate in tomato fruits.
Slide move. Well, good afternoon, everybody, and yeah, is it okay? Yeah, I think I'll just move on the. I'm more comfortable walking and talking. No. So, seeing the unseen, sometimes you look at plant, but you don't see what goes inside it. I think that I just talk about that. I hope it works. Oh, I think I do. It's weak. The wireless is not that efficient, so I have to stand here maybe. Well, the basic issue I'm going to talk about is about the hidden hunger. We all have sufficient calories, but we miss something which is vitamins and minerals in our diet, And we had got a major grant thanks to Professor Sapuri, which was related to biofortification of tomato. So it's not a problem which is in India, it's all over the global problem. So we have, 2 billion is something like 25% of the whole world population. But unfortunately, out of the 1/3, we are in this country. And if you look at that, if you don't have enough nutrition, it also affects the productivity. So your productivity, you calculate terms of GDP, when people are not report to work or they are not able to work, it is can be as much as 3% of GDP, and if you calculate in terms of the rupees, it is enormous amount of money. Okay, now hidden hunger can be addressed by providing a better food with the proper nutrition, and just shows that how much vitamin A deficiency is in India. And also, you look at the folate-related anemia in the pregnant woman. This is also one of the major part of the deficiency in our country. So, tomato is a good source for lycopene, beta-carotene, and as well as not for folate. So, if you look at both the calories, and we eat a lot of rice, but only one of them has sufficient amount of minerals and the vitamins. So we'll say no to this, yes to this, because it's a mixture of the vegetables, other things which will bring you the food. Rice will bring you only calories, and since you're using the polished rice, you're losing vitamin B12 also in polishing of the rice. So tomato is a good source, and we are focusing, I think I'm going to focus today more on folate, but we also have a little bit of the vitamin, lycopene coming over here. So question is that why do you want to biofortify tomato fruit? If you look at the vegetable consumption in India, potato is the largest vegetable and tomato is the second. So you can provide a cheap and alternative source, for the nutrition to the masses, and you can grow the fruit or tomato plants even in backyard in pots also to have a nutrition rich tomato. So that's the idea here, and I'm going to talk more about folate as well as lycopene in today's talk. So we look at the folate accumulation in tomato. This is the fruit ripening stages. Arun showed excellent pictures. Folate actually is related to cell division, cell differentiation. So maximum amount of folate get accumulated during when fruit is expanding and growing, and amount declines when fruit is mature. So folate peaks around the fruit expansion phase and declines. And if you look at the carotenoids, they accumulate, as you can see the color over here, when the fruits start ripening. So under the project, we evaluated large number of sections of the tomato, and we looked at more than 125 sections of tomato using mass spectrometry or the folate level, and you can see the folate is something like 10 microgram folate for 100 gram of tomato fruit fresh weight, and you get about 50. Some of these are wild relatives, so you have not much variation of the folate in tomato. You get only threefold. So we have we looked at the mutants which were isolated on lab and see what is the level of the tomato folate in these tomato mutants, and we found that cultivar we are using, Arka Vikas, has low amount of the folate, but we isolated one mutant which is trifoliate, had something like 80, so you are jumping from 20 to the 80. And Arka Vikas is one of the most popular variety grown in South India for the tomato consumption. It is a non-hybrid tomato, and it's cheaper, the seeds are cheap, so it's very resistant to drought, so farmers prefer to grow that. So this is somehow increasing, and that's what I've titled the talk as Seeing the Unseen, because you don't see what is inside the fruit. It's only when you do the extraction and the mass spec analysis, and then only see how much of that nutrient has changed. So this is a trifolate mutant. Look. I can just leave it. So this is a trifolate mutant. It's a mutant which was well worked on, and we isolated in our lab when we were doing the tilling project, the EMS population. Actually, the student was isolating non-phenotypic mutant and trifolate phenotype combined. So he segregated both the phenotype by genetic back crossing. So he made 2 or 3 back crosses and segregated out. And the other mutants which are from other labs, we collected all of them, because this is the one which is in the MYP117 transcription factor, and here it will make a truncated protein, because protein get terminated at this particular amino acid. And when you looked at the folate level in the fruit, normally in the tomato varieties, the folate level declines during the fruit ripening, but here it was not declining, it was stable. And when we made a process, we found that actually this is this is not good phenotype. The farmers may not like it, because this is with 3 leaves here, but it is semi-dominant, So you can, using it as a hybrid, you can probably increase the level of the folate. And we have checked each and every of these trifolate mutants, and we found that TF1 has a maximum folate, TF2 has, others do not have. That shows that how this particular transcription factor is affecting, depending on where exactly mutation is sitting in the factor. And when you cut it, the fruits, this is the Arka-Vikas, we found the fruit is very red. So it's not only has folate, it also has high amount of the carotenoids. So it also had a high amount of carotenoids, and if you look at the carotenoid level in the wild type, is progenitor, all total carotenoids were 107 microgram, and here you have 215. So this has a double things. You had high folate and also almost double amount of the carotenoids, and also a semi-dominant gene. And then we look at the metabolome, that's what we want to see, and we found that large number of the amino acids were going up in the mutant. This is the differential expression, that is whatever is showing is the wild type is 0, and then full expression is calculated from that, So that's the wild type will be always over here, and this log 2 fold, so it means we see 5 year, it's 32 fold increase in the metabolite level, It had high level of the amino acids, and one of the things recently made a news in the I think Nature Plants, and I think Hiroshi has done that. They generated and released GABA enriched tomato in Japan, and we found that GABA in this particular mutant 32 fold higher than the wild type. Only thing we have to estimate in physical term, the relative expressions, physical term, how much GABA is present in this particular mutant. Then we did transcriptome analysis. We are going for systems approach. We found large number of transcripts upregulated in the mutant, and these are all backcross line, which is backcross 3 times, and then BC3F3 or BC2F2, F3 lines, we did large number of transcripts went up across the different geo classifications. So mostly were upregulated gene and downregulated gene. Our whole idea was that can we go on using transcriptome and pick up where exactly the folate or carotenoids are increasing, and as you can see, folate is involved in C1 metabolism. The C1 metabolism also affected several transcripts related to this are affected in this particular mutant line. And if you look at, this is the MIB which is mutated in this, it is affected several other MIB expression. Wherever you see the brown, this is upregulated, where you see the blue is downregulated. So one MIB is affected, but MIBs are working as dimers, and they complement other MIBs also. So it is affecting the global expression of the several transcription factor. Okay, then we did proteome, and again we found large number of difference in protein. And if you look at the protein profile, there is a metabolic pathway where you can see different proteins working, amino acids, light reaction, photorespiration. Again, you can see C1 metabolism, nucleotides. As you shift from one stage to another stage, you see change in the protein expression also. So this is not static but dynamic. And transcriptome data which I showed you earlier was at the red rabbit stage of fruit. So transcriptome and proteome was significantly altered in case of this trifoliate mutant. The question that I showed you earlier, that what elevated the amino acids in the trifoliate mutant, and when we did the proteome analysis, and what we found that most of the amino acid tRNA synthesis were not getting charged. There was a significant down regulation of these proteins in the TF mutant. So that was the reason probably the amino acids were not directed protein synthesis to that extent, and that's led to the enormous increase in amino acid in this particular mutant. Sorry, I just had to go. For this. Oh, I missed which side I'm going, right? Okay, so our proteome analysis showed that there is down regulation amino acid tRNA synthesis in the TF mutant. Now, question was that, see, I showed you earlier these pictures with the mature green breaker, how the overall global proteome was affected. But then you have to go and look at specific proteins, what affected a particular pathway. And two things we found over here, one I am showing only here, two of the proteins which are involved in the carotenoid biosynthesis, particularly lycopene, were upregulated. You don't see upregulation transcripts over here, because transcriptional studies shows down regulations, many of them, but these proteins are upregulated over here. In addition to it, it's not only these proteins, several proteins which are involved in the holding on these carotenoids, sequestration protein, they are also upregulated. So carotenoid synthesis probably came because of upregulation of this protein as well as the efficient sequestration of the lycopene. Now question was that what upregulate folate in trifolate fruit, and we did transcriptome, we did not find any change in transcriptome. When we did the qRT-PCR, we got actually some results, but at red dry stage we got maximum folate, we don't see much of the change in the transcripts. But when we looked at the folate degradation, we found two of the enzymes which are involved in the folate degradation, gamma glutamate hydrolase 1 and 2, are down-regulated. And then we went and checked activity, we found the activity of these two enzymes were much lower into the trifolate mutant, and that was the reason why the folate was upregulated in this particular mutant. Okay, so that was, we ultimately landed on some place where we see the folate was upregulated. And of course, as you can see in this graphic, that from when you have mutations, the wild type gene, the mutated gene, this part is missing. You have high lycopene, high folate, and high gamma amino butyric acid. Just continue a little bit more. We also, as I showed you earlier about the examine 125 accession of folate in tomato fruit, we picked up, we compared two local cultivar. This is the cultivar grown with, in again South India, FKM1, released by Tamil Nadu Agricultural University and Arka Vikas. This is low folate one. We found this is high folate. And when looked at the folate, it is slightly higher folate than this one, Arka Vikas. And then we examined here also what happens to carotenoid. It also high carotenoid. So we are finding in our lab, whenever folate goes up, the carotenoid also goes up. So there is some relationship between the two of them. We again went and looked at the same gene expression. We found that some of the gene expression for the red ripe stage were lowered, and also had a low amount of the gamma glutamyl hydrolase activity was also lowered. So then we went and knocked away two of these glutamyl GGS genes. I have not shown the data here because they are not published yet, but we found that when you knock out the GGS genes in tomato, GGS1 and GGS2, the folate level goes up by threefold. So we had made the knockouts of that, and we had high amount of the folate level. Well, I think this is the end of story, and this is we have the papers which we published out of this work recently. This Spiraculum tomato came in horticultural research, and then the title which you see, Seeing the Unseen, came in plant journal this year, and this was the starting point for this work where we did the natural variation in the folate level in the tomato. So, I think this will be the final slide. What I want to say that when you are progressing in the journey of your life, somebody is taking care of your growth and development, and that person was their mentor. Happy birthday, sir. The birds. Thank you, sir.
Thank you, sir. I would like to request Professor Sapori to give the token.
Thank you, sir. Thank you.
Very great. Centered.
Yeah. I should thank Bhai for supporting the center, because he is the one who granted me the building for the center. Sir, you are the chairman of the committee. Thank you.
So that brings us to the end of the second session, and we'll break for lunch now. And since we overshot time in this session, so request everybody to be back by 2. The lunch would be in the guest house, so there would be volunteers to show the way to people who are new here. Thank you.